Good morning. I know that the morning might be tough, at least for part of the consortium, but it's my great pleasure to welcome you again. Today it's a third and unfortunately the last day of the workshop, but I think it's great and we plan to make more workshops. So Warsaw team, be aware that I have more plans to do next workshops. But first, we will follow Eva's recommendation to relax. So first, we will just relax. Okay, as you know, yesterday we were recommended by our vice rector for science to enjoy Warsaw, relax, and to go to the pub. So we follow the orders. So the team might be sink in very well now, but probably a little bit tired. But we hope that this day will be also full of very thoughtful discussions, and we will make our best to make it very interesting. So this is the agenda for today. So we will start with very interesting lecture conducted by our partners from University of Trento. So the team from Trento is responsible for signal processing and all the computations. They are very experienced, so we are really looking forward to their talks and then for their workshops. So this talk, Introduction to Interpersonal Synchrony, will be followed by the talk of Professor Tomalski. He's head of Baby Lab in Polish Academy of Sciences, and it's also my colleague. We have known with Przemek since several years, I think. We also conducted studies together, including FNIS, because Przemek is an expert also in applying multimodal techniques to study baby development, child development. And again, then after the Przemek's talk, we will go back to synchrony, and then we will talk about interpersonal synergies in social interactions, from parent-child play to dance improvisation. And then again, we will dive into signal processing, and then Professor Kmita from Faculty of Psychology, Warsaw University, will give us a talk about bio-behavioral synchrony in early interactions of parent with preterm babies. So I am also sure that it will expand our knowledge. And after the talks, we will have workshops focus on signal analysis. Okay, so I think it's time to start. So Alexandro, the floor is yours.
Okay, yeah. I switch 1 second the presentation. No, I think it's, yeah, should be here. How do you... I think the screen is mirrored. So you can see the... How do I put it on a different...
This mirror, what do you need? You need
to put in a different part of the
this
notes.
I'm not sure.
Otherwise it's fine, I do it without.
Yeah, you can do without, or maybe. I think with Google Drive you cannot do it.
Okay. So hi everyone, it's nice to see a lot of you after the dinner we had last night, and this is very early in the morning today. Today, to wake us all up and to help facing the day, I will talk a bit about theories behind interpersonal synchrony, So what we know about synchronization between people at multiple level of analysis, so the behavioral, the physiological, and the neural level of analysis. Okay, so I have to do a disclaimer before we go on with the presentation. Today I will present mostly studies on parent-child interaction, but of course interpersonal synchrony is not limited to that specific type of dyad. But I took this choice because, of course, the concept of synchrony was initially developed in the mother-child, caregiver-child interaction to assess, let's say, the quality of these exchanges. Of course, we know that early interactions are crucial in our life. We, since the beginning, we learn through interacting with others, and the interaction with the caregivers at the beginning of our life, it's particularly important for scaffolding social and emotional development in the infant. And then longitudinally, we can say that the quality of the early interactions has an effect on the subsequent attachments throughout our life. So when we experienced high quality interactions in the early phase of our life, we tend to, let's say, keep this type of quality and the patterns that we observed in the early interaction throughout our life. I said many times the word quality, but how can we define quality? And how can we study quality in a social interaction? When we study an interaction, we have a series of problems because we are not studying one person, we are studying two people that are interacting with one another. So the first thing that we can do is to switch from studying the two individuals individually to study the dyad as a unit. The second thing that we can do is that take into account that both partners are able to influence each other throughout the interaction. So both the caregiver and the child are able to regulate this interaction. So we can consider the dyad as a dynamically interacting system. And the third point is that we can specifically focus on the exchange of information between the dyad. So the way in which the information flows between the interacting partners. So we have defined how we can study a dyad, but what do we look in an interaction? So there are several concepts that have been developed in developmental science, such as the concept of freedom, so the temporal structure of the interaction. Think about the typical structure of a conversation, for instance. We have a turn-taking between people, and we have a temporal pattern that a conversation should typically follow. We have the concept of reciprocity, so the adaptation of the partners to each other. So for instance, the mother that smiles and the child that smiles back to her. And of course, last but not least, the concept of synchrony, which means the alignment in the temporal structure of events between the two interacting participants. So a few years ago, we wanted to understand how, because as you know, the literature on synchrony is very wide, there's a lot of studies on it. So we wanted to understand its structure. So what are the most commonly studied topics in the literature? And we adopted an automated approach on a large-scale data set. So we basically downloaded all the available literature on synchrony, mother-child interactions, which were more than 500 studies. We looked at their references, which were about 29,000 studies. And then we adopted automated techniques to cluster the literature based on how close these references were in the citations list. And what we observed is that the literature moved, let's say the literature took 3 steps in synchrony. And it moved from early studies on behavioral synchrony to studies on physiological synchrony, so the red group that you see here in the middle, to more recent studies on neural attunement, so neural synchrony. So these 3 were the, let's say, largest and most common topics of the literature. So that's what we were expecting to observe, right? A trajectory from behavioral through physiological, and then more recently neural alignment. So I decided to use that as a structure for today's presentation. And I will briefly present each level of synchrony that we have in the literature. Of course, these are not the only levels, because more recently there is, for instance, conceptual synchrony, so the way in which 2 people can align in their thinking, in their more, in their speech, so in the way they think about the world. Other studies show, for instance, alignment at the hormonal level. Now for today, I will focus only on these 3 levels of interaction. So the behavioral synchrony, we typically study through analyzing matching motor activity between participants. So people typically extract like the pose of participants and see analyze the alignment between participants throughout the interaction. Another type of behavioral synchrony that is very widely studied is the matching of affective displays. So for instance, the mutual smile between the dyad. And what we observe in the literature is that, for instance, when people align in their motor activity, they also tend to like each other more. So there is an effect on the likeableness of the interaction, so how much people like to be with the other participant. There's an effect on the prosocial behaviors. So the more people, even unconsciously, align their motor activity, and the more they're likely to have prosocial behaviors with the other person. And then we went through the phase of physiological synchrony, which is not a phase, I mean, we still do it very often, and which refers to the matching of the autonomic nervous system activity. So the autonomic nervous system, of course, innervates many organs, and we can study this matching by, for instance, monitoring sweat glands, respiration of heartbeat of the participants. And there is physiological synchrony has been shown to relate with many behavioral outcomes, such as in-group and out-group bias, pro-social behavior again, how much the person likes the interacting partner. And there is a very recent meta-analysis conducted by the group of Ilanit Gordon that shows a positive effect of heart synchrony on relationship outcomes. And then at the end, we arrive to the neural synchrony. But why did it took so long? Because there was a very long gap between behavior and physiological synchrony to arrive to neural synchrony. And the fact is that if you think about the traditional paradigms in social neuroscience, there was no way to use them to study neural synchrony, because they typically involve one person, typically exposed to social stimuli, such as faces, voices, and so it didn't really mimic what we observe in our common daily interactions, right? And so for this reason, there was a boost, let's say, in the research of neural synchrony by the introduction of the hyperscanning approach. So the simultaneous recording of the brain activity between individuals, which was introduced in 2002 with this commentary by Montag et al. And you can see how how complex it was at the beginning to run an hyperscanning studies, because we had to synchronize two fMRI, at least in this paper, they had to synchronize two fMRI machines, which I think were on different sides of the world, with participants interacting with each other through by staying still and laying supine in the scanner. Of course, the hyperscanning literature took a lot of, has moved a lot from that initial setup. So for this reason, we very recently conducted a literature review based on an automated approach as we saw before to, let's say, identify again the most common topics of research in the hyperscanning literature. And in this case, we started from more than 500 articles on hyperscanning and conducted the analysis again on their references, so more than 30,000 articles. And by using clusterization techniques, we again observed many topics, even more than before, of research in the hyperscanning literature. And I wanted to highlight mostly two things. So we observed in the topics that while initial studies were mostly on EEG, there was a huge proportion of recent studies based on FNIRS, as we were saying yesterday from, I think it was Franzi showing the same data. And this use of FNIRS boosted from the initial study by QE and colleagues that we saw yesterday on cooperative game. But why FNIRS? Well, because the social interactions have a strong component of movements, right? We interact with each other by moving, by synchronizing our motions, grabbing objects, and so on. So we needed a technique that allows us to have these kind of social interactions, not social interactions that were with two people that were laying supine in a scanner and couldn't really move. They were just pressing buttons to see, to conduct a task. So another trend that we observed in the literature is this switch from highly controlled experiments that were conducted with EEG at the beginning, where they asked the participants to purposely imitate each other, to take a leader-follower role, to highly naturalistic experiments. So experiments where we see parent-child interacting, free playing, and moving around the room. And this was, of course, possible only with a technique that allows us to the signal not to be so compromised by motion. And I wanted to bring you an example today of what we consider a naturalistic experiment in the context of hyperscanning, which is not about caregiver-child, but is based on close friends. And it's a study that we have very recently published in Neuroimage, and where participants were free to interact verbally with one another. So we asked them to use a data set that we collected in previous studies that was very rich in dialogues. This was done to understand how the things we say throughout the interaction, the way we say the things, the way we structure our communication with the other person has an influence on the degree of neurosynchrony that we observe. So we had a data set with 3 conditions, which are not really relevant for the current presentation. The important thing is that they were all based on 5 minutes of dialogues. So we had a data set that was very rich in verbal interactions. We had 84 participants, so 42 couples of close friends. And what we did is that we video recorded the interactions, we manually transcribed them all, and then we applied automated techniques to extract 2 types of information from the dialogues. So the emotional content, so how the we had 8 emotions derived from Blue Chicks theory, and we had a degree of how much each emotion was expressed through in that dialogue. The second type of information was the semantic and syntactic structure of the dialogues. So the way in which participants were structuring their dialogues at the semantic and syntactic point of view. So how complex, how many, what kind of semantic aspects were they exploring, and so on. Throughout the interactions, we also used F-NIAS hyperscanning. So we monitored the participants' prefrontal activity, both participants' prefrontal activity. And what we did to compute brain synchrony is to use the wavelet transform coherence between homologous regions. So we had region, for instance, superior frontal gyrus of participant A, and we matched it with the activity of the superior frontal gyrus of participant B. And this is something that will come out very often today in our presentations, and also in the hands-on part of the workshop, to have a control condition and to understand which brain region showed it's a significant level of brain synchrony. We included we compared true and surrogate dyads. So true dyads are the ones that interacted with each other throughout the experiment. Surrogate dyads are real participants that took part in our experiment, but they were matched without really interacting with one another. So we had one participant from a couple that participated on day 1. We matched his activity with a participant from day 10. So these participants never saw each other in the experiment. And we did this because we basically want a control condition of participants that were involved in a social situation, but they never interacted with one another. Because of course, the level of synchrony we observe might be due to the fact that we just have people involved in the same social situation, not of the fact that people are really interacting with one another. And what we observe here is that 3 regions showed a significant effect of true versus surrogate dyads. And these were the superior frontal gyrus and the bilateral middle frontal gyri. And then we used the emotional metrics that we computed before, and as well as the semantic and syntactic metrics to predict this level of synchrony in these 3 regions of interest. And what we observe is that the emotional content was not really able to predict synchrony in one region of interest, but was more able to predict at the global level of analysis, of course, because emotional processes are not so localized in the prefrontal cortex. And the opposite pattern was observed from syntactic and semantic metrics. So these metrics were more able to predict synchrony at the local level, specifically in the right middle frontal gyrus, which is a region that typically is involved in these type of processes. So to conclude, why should we study synchrony at the end? So there are several studies that show that synchrony, at least in mother-child interaction, is predictive of cognitive development. So for instance, processing of stimuli, of language development, and even IQ, I think. Of adaptation, so the way in which the child adapts to the surrounding environment throughout their life. It's a concept that depends on both partners. So both partners are involved in this process. So we are taking into account both partners when we study synchrony. And at the end, we also observe, in fact, that synchrony depends on the caregiving skills of the caregiver. So we observe that caregivers that are less intrusive and more sensitive show also higher synchrony to their children. However, it's not so easy because synchrony often is associated with outcomes that we don't really expect, such as here. This study was published last week, I think. And they showed that synchrony was associated, higher synchrony was associated with sexist comments, which is not really an index of social connections, right? This to say that, I mean, it's hard to interpret synchrony alone. We always need to take into account, I think, the context in which synchrony is, we observe that amount of synchrony, what at the behavioral level, at the environmental level. So what is the context in which we are observing this type of synchrony? And thank you all for now.
Thank you very much. Don't run.
Okay.
Do we have questions to Alexandra? Questions? Yeah. Oh. Yeah, we were encouraged to use the mic for questions.
Thank you for this very interesting talk. I was wondering because something that you said struck me that when you talk about physiological synchrony, you actually talk about the autonomous nervous system. And for example, we know that in neurodevelopmental disorders, we have people who cannot interpret their autonomic nervous system as well as others. They have different interoceptive abilities. So are there any studies that link interoception with synchrony?
I think, so there are a lot of studies, and in fact, this is, oh, I mistook the year, of course, it was 2017. But you can see that from our research, there is a strong, there's a group of research that specifically focus on interoception and synchrony. And it's very recent. I have to admit I'm not really up to date to that specific topic. What I would imagine is that, I mean, of course, there are people that are, as you said, have challenges interpreting their own autonomic responses. And I would imagine they, so what is the prediction that you would make when we study synchrony with, in these populations, for instance?
Well, one of my ideas, and this is a very far-reaching idea, right, is that basically when we interact with the world, we tend to include the part of the world that we interact into sort of our peri-body space, right? That when we have a tool, we actually can see the representation of the tool in the cortex. And that when we interact with the other person, we also include this person in this our peripersonal space and in our, we have this idea of this dyad also in our perception. It's not just that we have this person that we interact with, but we also have a representation of this person. Yeah. And that my wild guess here would be that when we are, when we have people that differ in their interoceptive ability, they might also have a different representation of the other person in this peripersonal, this interactive representation of this interaction.
Yeah, I think so. So in this interpretation, I think it will be very interesting to adapt it to synchrony studies, because of course the way in which we are able to mimic the other person's responses at every level might then be influenced by our capacities to let's say represent them at the mental level, at our cognitive level. Yeah.
Thank you.
So I think so. Yeah.
Other questions? No. Okay, so thank you very much. We have also a small gift for you. It was a great talk. Thank you so much. Okay, so now we have a second talk by Professor Tomalski. So let's switch the computers. And I will repeat again because a few people were late. So Professor Tomalski is the head of Baby Lab at Polish Academy of Sciences. He's a leader of many projects funded by our Polish agencies, but also European Union. He has an experience with applying various techniques, including FNIS, EG, motion tracking, vocal tracking, probably more behavioral techniques. Very experienced researcher. So yeah, Przemek, the floor is yours. And thank you very much for accepting our invitation to this event.
Thank you. Well, thanks very much for the invitation. Can you hear me well? Yeah? Okay, perfect. So it's a real pleasure to be here and to look more at the recent advances in neural synchrony in social interactions. I have to admit I do not have a single slide that there's a shred of brain data today, so I'm going to focus on behavior, and for very specific reasons. I'm going to talk about both motor and vocal aspects of early social interactions, coming from a slightly different angle. So not to confuse you too much, I'm going to talk about coordination rather than synchrony, because I will be focusing on the things we do rather than things that happen in our brain. I should also mention that a lot of this work wouldn't be possible without wonderful PhD students and postdocs in our lab at the Polish Academy of Sciences, especially Zuza Laudańska. You'll see the contribution. Also, Asia Duda-Gławska, who is also collaborating with Agnieszka and Agata Kozio. Okay, so the story of how social interactions actually serve as the basis, the medium of development, or are providing mechanisms of development. These stories have different origins in different literatures, and it's actually quite confusing when you start reading different literatures, trying to piece it all together, because there's one line of research which was also cited in previous talk, coming from people like Ruth Feldman, clearly inspired by Tronik's work with the still face paradigm, with looking at face-to-face interactions in a very, very, very specific period of life, around 4 months of age, but not later and not younger, usually. And also inspired by a lot of sort of psychodynamic literature. There's other strands of work coming from ecological psychology, people like Karen Adolph, who study motor development in sort of various ecological environments, also moving into interactions. There are people coming from dynamic systems like Linda Smith, who have their own programs of research. All these people look at social interactions in a completely different way and focus on different mechanisms. So how do we sort of piece it all together? Well, I very much like this paper from Vasu Reddy from 2003, which may misleadingly talk about, being the object of attention and kind of development of self-awareness, but do not be alarmed. This is actually very much paper about trying to put together a coherent idea for why we coordinate behaviors with other people, and what are the actual skills that may emerge out of social interactions where we're encouraged and sometimes forced to coordinate with other people in the first years of life. It's usually our caregivers. So what I'm showing you here is that there's also a whole idea of how you could ground different skills that you can see in this table by talking about infant responses to and actions upon other person's attention, by either responding or by directing attention at different ages, and that's related to sort of potential developmental changes in the aspects of the other person that the infant is able to track, to follow, to attend to. And what you can see here is that there's a very nice progression of different skills that increasingly focus on different modalities. They start to involve more modalities. And when I look at this paper 22 years on, I'm amazed at how nicely, albeit slightly implicitly, it shows that your different skills across the first couple of years of life in coordinating behavior are very much limited by capacities of your body and by your sort of low-level cognitive capacities, like attentional skills, redirecting attention, following gaze, like being able to attend to things because your head is actually able to move freely because your muscle strength has increased in the first half of the first year of life. And then how you're able to establish more complex aspects of interaction because you're able to handle objects in your hand, finally, sometime in the middle and in the latter half of the first year of life. And that whole situation is suddenly inviting your parent, your caregiver, to comment on the objects, to talk about these objects, to produce a lot of very complex social situations just because you're able to sit on your bum and you're able to hold objects and manipulate them in your hand, okay? And those biological bodily constraints are very much underlying those cognitive skills that you see here. And what I've been very much curious about is how in recent literature has sort of caught up with this whole problem of how bodily constraints actually sort of define and limit what we can do in social interactions, and that we rather poorly understand the grounding of certain skills in basic development of motor skills, of attention, of speech production. And one nice piece of literature that I've seen in recent years was this chapter from the book called The Pragmatic Turn, which is talking about action-based frameworks for studying cognition. In particular, coming back to this existing in the neuroscience literature idea of sensory motor contingencies, which is going back to sort of active perception accounts, where you actually, when you're perceiving stuff, you're as much creating it in your brain as you're just receiving information. And the fact that your perception of objects and space is tightly linked to your bodily activity within that space and on those objects. So what we perceive is as much a product of handling an object. So imagine I'm holding a ball in my hand, and my perceptual systems very much are defined and limited by information, the interoceptive information from my hand about the object I'm holding, and by the fact that I'm rotating and moving that object in my hand within the space, including peripersonal space, as being noted in the question before. So that there are these sensory-motor contingencies where I manipulate objects, and what I get perceptually back is, in a way, the product of my action, okay? So what we can say is that also is that social interactions can be conceived as this sort of set of sensory-motor contingencies, right? Because you have actions directed to another person, so you have to be aware of that person, but then you have to be able to produce specific actions that will have certain consequences for that other person so that you get something back, okay? So there's a whole very interesting, now slowly but surely growing theoretical literature stemming from this piece and others that is trying to understand social interactions as a set of sensory-motor contingencies where you have certain biases for perceiving certain things and not perceiving other things, like biases for attending the eyes, and certain constraints that perhaps come from your brain or from your cognition. Okay, enough theory. I've spent far too much time on that, but I want to tell you that increasingly I've been trying to get together to multimodal social interactions by looking at different modalities. So we've done some work first some years ago with a former PhD student of mine and Maciek Haman, with Alicja Niedźwiecka, looking at very simple instances of attention coordination, just measuring the amount of looking into each other's eyes around 5 months of age, demonstrating that this dyadic coordinated activity, albeit very simple, actually predicts specific attentional skills measured with eye tracker in the infant 6 months later, even when you control for individual differences in that particular skill from previous age. So we have a very nice tight control here. And that just simple duration of mutual gaze, so looking at each other's eyes from 5 months of age, explains around 8% of variance in attention disengagement in a very kind of standard gap overlap task. I won't go through much of detail. Sorry, if you have any more interest in these tasks or details, ask me a question later. So my argument would be that social interactions serve as the context for most learning. And one nice thing that you can see, and people haven't looked at this for a long time, and we got very interested in the lab at some point about this, is that they permeate even sort of very constrained experimental situations. So look at that very clever thing that this mother is doing with redirecting attention of that baby just by grabbing the arm, and moving, there you go again. And this is happening completely spontaneously. No one has given any instruction. The instruction was, in fact, do not interfere with the task. The task is a boring habituation task. So, naturally babies look away from boring stimuli. And this mom just spontaneously was doing what she thought is the optimal way of redirecting attention. So even when we are trying really, really, really hard to do constrained, controlled experiments, social interactions, at least in infants and young children, are there, and there's no way to get rid of them. So we started to think about some other ways of looking at this. So there's just a paper that may serve as a starting point, albeit it was it was a social interaction observation study of situations when infants sit on the parent's lap or in a high chair, and they do eye tracking experiments, very standard ones. So we've annotated tons of different behaviors, and then we've started to look at differences in parental visual attention, differences in touch. There is a coding scheme that we've also published. I think we would have done things slightly differently nowadays, but I'm very happy to see this published a few years ago. So we even started to argue that infants are a sort of a dyadic learner. So, it's really hard to cut them off a social interaction and study as an individual as such. And that is a serious problem for most of our experimental literature on cognition and cognitive development that we have. Some of the findings in that particular analysis of a fairly large group of 5-6-month-olds were showing that caregivers differentiate their looking at the screen depending on the eye tracking task that the baby is doing. So they implicitly respond to differing task demands, of what their infant is doing. Parental behaviors differentiate even habituation conditions when there are more engaging, interesting, and boring simple task conditions. And caregivers' physical contact differentiates eye tracking tasks, and there are also associations with task performance. So, for instance, some parents produce more active physical touch during boring habituation trials, and that is related to looking durations. Okay, so it does affect task performance. Okay, so these are examples of the fact that we find social interactions everywhere, and for a long time they've been focused on visual attention. There's also been separate literature on dyadic vocal turn-taking that's been developing for a couple of decades. But we've been trying to put together the vocal and the dyadic story. And one recent set of studies comes from our longitudinal study, which we've conducted. I'll talk about it in a second. The key author here is Zuzanna Laudańska, who is a recent PhD graduate. Dr. Laudańska is, funnily enough, now in Heidelberg, although in a different team. So at the university hospital with Peter Marshik and his wife, Tassia Zhang. So what we've been trying to look at is inspired by this. I'm not sure we have sound here, but maybe you can hear. Can you hear it? Yeah, I think. Oh yeah, yeah. Uh-huh. So the point is that the mom is talking, but there are also leg kicks of the baby in response to talking. Okay, it may sound quite boring, because there isn't that much vocal turn-taking. Obviously, this is a young 5-month-old baby. But the point is that there's a lot of other motor action going on, other motor production from limbs in particular that are going on in association with vocal production. And right now we're in the middle of analysis that are trying to look at both vocal and motor actions together to see whether they're at some early stage of development equivalent or kind of placeholders for each other in a sense. But what we focused on in our studies was trying to get, as previous talk indicated, from rather constrained situations. So here, top left, you see a set of very classic designs from Linda Smith's lab. 10 years of fantastic papers year in, year out, looking at babies from 9 or 10 months of age onwards up to about 12, 14, using head-mounted eye cameras and eye trackers, having 3 objects, white coats, white table, looking at vocal and attention production and how infants interact with objects, coordinate gaze with the parent, shift attention, lots of fantastic work, but what you notice is the fact that they are seated in front of each other. Go ahead, if you want to sit down here. It struck me only when we started to try to do stuff, in more kind of semi-naturalistic setting on the floor, that there's just so much wonderful work, but it's so constrained situationally. And there's very little developmental literature that would be able to use more sophisticated measurements of behavior in unconstrained settings, because there's just so much noise, so many extraneous movements, behaviors, and most of all, that interactions are a messy thing. You know, if you're a baby or an adult, social interactions are messy and quite unpredictable in many situations. And it's a constant problem with trying to measure things on a time series where you just don't have enough behaviors to analyze coordination because they are rather spurious and infrequent. So what we've been trying to do, struck by, how constrained the Ewan Smith situation is, so I should mention Chen Yu apart from Linda Smith in all those studies. So we've been trying to do different pilots, as you can see, with bouncers and with wearable accelerometers, with some other gizmos as well. Discovering that seated babies are not the most socially interactive babies either. So we've moved to the sort of as unconstrained as possible situation, instead opting for constraining behaviors, not by seating, but by the type of activity. And it proved to be a very intriguing sort of semi-experimental manipulation in a sense, but still leaving a lot of freedom in our dyads to do different things. And we started to do analysis where we actually systematically compared vocal and motor activity across those different activities. So book sharing, which has a lot of vocal production on the parent side, but also coordinated looking and sometimes even pointing in older babies. What we call manipulative toys, so different kinds of toys that afford a lot of interesting manual actions on the part of babies, less talking, less coordinated looking, parents may be slightly less involved, and rattle game, which is again the sort of interactive game which involves a lot of reciprocity. There's also a lot of attempts at coordination because you're trying to rattle with your parent. There's also a lot of singing. So we have a lot of activities where we try to squeeze as many vocalizations out of babies, frankly, within 5-minute sequences. So we had those, but they were very different in task demands. So I'm going to focus on those three at the top. And we've done exactly the same set of games, maybe with slightly different toys for that are appropriate for younger or older babies. We've seen this large group of babies at 4, 6, 9, and 12, and then they came back for certain outcome measures at 25. I won't have time to talk much about the wearable sensors today, but they were there, and I will show some data in a minute from it. The point is this, that we've been looking at both the individual level and the dyadic level. And the intriguing thing is that when you start to compare systematically those different games or tasks that are still semi-naturalistic, you start to see very intriguing differences in what people do. So here you see rate per minute of caregivers' vocal production, so how often the caregiver was saying something, and you can see that the caregiver is super constant and remarkably kind of predictable in looking at those different tasks. So during book sharing, there's most vocal production in the blue bars, and during rattling, there is less, as well as during manipulative tasks. So the parent is very systematically from 4 months of age doing the exact same thing. During book sharing, lots of production, lots of vocal production, and much less during other games. On the other hand, when you look at babies, you can then look at the emergence of those task differences and how they actually learn to adjust their production, their activity in the vocal domain to the task. So what you see here is, I'm not going to be showing the stats, but this is multi-level modeling. You can see the paper for more details. This is, again, the rate per minute of infant speech-like vocalizations. So they were all annotated manually in line with the existing literature on what constitutes a speech-like as opposed to reflexive, cries and laughs and so on. So you see here only those speech-likes. And you can see that those kind of task or game differences only emerged by age 12 months. Intriguingly, there's as much production during rattling as it is during book sharing. But the very exciting thing that we see when we actually look at conversational turns, so turn taking. Here we have a number of turns because this is a constant timing across our dyads. But if we calculate it into rates, you would see similar patterns. You can see that these turn-taking rates emerge on the dyadic level earlier than on the infant level, okay? So you can see book sharing producing more conversational turns between the infant and the parent already at 9 months of age. Earlier than we see systematic infant behavior in terms of the amount of production at 12 months. Sorry, this is, earlier 9 than with an infant. So what is interesting for us to see here is this sort of very slowly emergent pattern of adjustment of the vocal production system to those different task demands. And then we got very, very curious about the motor system and how it may organize to those specific task demands. And the interesting thing is that when we try to look with various measures at motor activity, specifically limbs, so legs and arms, we saw a fairly similar pattern of very slowly emerging task differences in limbs by 12 months of age, very closely mimicking the vocal domain, showing that there's a lot of parallel between the vocal system, and which is a motor system as well, and limb activity. So I'm going to start with what you could consider sort of task-related flexibility or kind of complexity of the system, just to show you quickly a bit of info. So we've tried to play in this study, and I think to a large extent successfully, although it wasn't easy, with wearable accelerometer sensors that are more advanced than kind of super simple ones, for the reason that they were lightweight and wouldn't constrain movement during social interactions, as well as we could reliably synchronize and measure with one kit the activity of the parent and the baby. So there were clear advantages to this. There were also some disadvantages if you're interested in ask me, especially in synchronizing to vocal. But Asha produced a very nice demo, I think, video here showing the first top 4 lines are infant limbs, and the bottom line, I think, is parents' arms, left, right, yeah. So you can see that we get actual precise measurement, albeit we don't know what that movement is, but we can actually apply some complexity measures like recurrence quantification analysis to this data to look at entropy of movement. And this is what we did. This plot actually comes from Zuza Laudańska's PhD thesis, but various parts of that data you'll find in those 2 papers. When we looked at entropy, which you can sort of consider as motor system complexity, this was a useful measure for us to look at how these different tasks organize motor activity in different ways, showing that there are also very similar, slowly emerging patterns of motor system complexity only by age 12 months, also showing, manipulative task producing different level of complexity than those tasks that produce more kind of concerted, coordinated activity. a similar pattern is if you kind of zoom into more kind of lower level movements. So we've also looked at rattling games specifically because we were interested in learning how the bimanual coordination, so left and right hand during rattling, are actually coming together or not. And here we've looked at obviously some basic metrics that show increases in the number of movements and frequency during rattle shaking game with a parent. But the interesting thing is when you look at wavelet coherence of left and right arm movements, and I should mention that we knew that these are rattling movements from the accelerometer data because they were all painstakingly manually annotated by Zuza and students. So each one of them was manually annotated from the video so that we knew that we're not analyzing mess in those hand movements. And you can see that there are those very interesting systematic increases where motor system is very slowly catching up with different tasks and being able to produce them in a kind of systematic way. And you see those increases systematically by age 12 months from around 6 months of age. So between 4 and 6 months of age here, you don't see differences in coherence between left and right hand rattle shaking. And then there are systematic increases by 12. Okay, it's the same. Interestingly, we've been asked by the reviewers to look at effects of maternal rattling or singing on different metrics, and we haven't seen any effects on that. So interestingly enough, there are certain motor actions that are so difficult within the first year of life that you're still so focused on figuring them out that you're unable to participate in a kind of more sophisticated, coordinated play in a regular fashion in this case. So, for instance, we haven't seen any effects of mother producing or providing some sort of rhythm or not. If you ask me what these recurrence quantification analysis RQA derived entropy measures mean, we've also been able to try to look at some sort of individual differences in interpretations. And in a recent paper in IBD, and this is, I think, going to be my final bit, it's quite interesting to see. And this is actually a pre-registered study, so I'm very glad we did that pre-registration. It sort of went the opposite way to what we were expecting in terms of direction, but I was quite sure I will see associations between negative affect from a very kind of common infant temperamental questionnaire, but no associations with surgency, which kind of you could translate into extroversion. Nor from orienting/regulation. So these latter two subscales or constructs do not correlate with movement entropy from wearables, but infant negative affect it does, and it does so, as you would expect, in the same fashion across all three tasks. So there are no task effects or game effects on this association, okay? If you have higher negative affect in your temperament from your infant behavior questionnaire revised, then you have higher movement entropy across all tasks. Interestingly enough, we've also added, because we like to control for this, maternal trait anxiety with SDAI, so this very common quick state trait anxiety inventory. And at 12 months only, but not earlier, you get to see the effects of, or associations, I should say, of maternal trait anxiety with movement entropy in the opposite direction, which is something I'm unable to explain. We're still trying to look at this. But curiously, if you look at this plot carefully, and remember the levels of parental engagement in terms of vocal production, it was higher for books and then to some extent for rattling, and lowest for manipulative. And here we, in multi-level modeling, we see that maternal trait anxiety does associate with entropy of infant movement, but only for books and rattles, not for the manipulative tasks. So you can see that logically, tasks that engage parent more or perhaps invite more to coordinated play are more affected by differences in maternal trait anxiety. Okay? So this is very interesting. We'll be obviously following this up later on. I'm unable to tell you much about the 2-year outcomes because we're still in the middle of analyzing them. However, I think there's a very interesting process going on here whereby we see very close associations between the vocal domain and the motor domain. And specifically, this interesting fact that vocal turn-taking may be sort of driven by parental activity in different situations or specific contexts where, you know, there's this interactive engine going, producing a lot of speech, but also producing a lot of situations where you can respond. I haven't mentioned, but it's in that paper about vocal turn-taking that book sharing also produced the most stable and longest gaps between the parental speech and the infant response. So there was also much better temporal structuring allowing for this vocal turn-taking to happen. Okay, so we see those stable task differences for parent, but infant motor system and vocal systems are catching up very slowly, only by 12 months of age. We see kind of similar pattern looking at the very high level with RQA and entropy and sort of lower level with arm coherence of motor system. The data I haven't been showing you today, but we're currently also analyzing is we're looking at this potential idea of limb movements being early on a sort of a placeholder for emergent vocal turn-taking as well. So please watch this space in the future. And also there are some interesting results showing individual differences, which obviously may in the future have some clinical implications if followed up with other studies for sure. So I guess what I'm trying to say in my entire talk is that coordination of different motor systems for producing actions is a very slowly emerging process, at least across infancy, and I'm pretty sure that that goes beyond up to the preschool age, knowing what we know about motor control. And in the fact, then, the first years of development of social interactions are very much heavily constrained, not just by cognitive skills of the infant, but kind of very basic motor production issues, posture issues. We now are exploring a lot of postural aspects also with Jarek and Asha. So there's a lot of interesting questions there. So there are a lot of very low-level biological constraints, which I think we often forget when we study social interactions in first years of life, that are very interesting to explore and to better understand, especially when you move to the brain level. And obviously there's a whole story between motor and brain, which I'm not touching upon today. Thanks very much.
Thank you, Przemek, so much.
Thank you.
Questions?
I have a question.
Oh, perfect.
Mm-hmm.
How did you deal with the individual differences of participants? Because I'm also a parent, and I see that there are a lot of common issues, especially with children in this young age, that during the experiments they have so much personal differences that might actually be a bias or affect the whole process of experiment.
Well, you can see individual differences in all the plots. And I think the most probably intriguing are those when you look at, for instance, at entropy. These are fairly large individual differences, albeit, you know, the whole point of doing these different tasks was to reliably measure within a single session 3 sort of 5-minute games analyzed here, whether infants are able to produce within infant difference. So that was our way of dealing with individual differences, just kind of doing a quasi-experimental design that is still semi-naturalistic and unconstrained. Obviously, the problem that you also mentioned is posture. There are huge postural differences, so obviously we get asked in some analysis about postural differences or variability of those effects, so there's a lot of work we're doing also with Jarek on trying to look at that. Also now having a fantastic automatic classifier of infant posture from wearable data. So I think we can break down into certain, constraints. I think posture is the biggest problem, other than achievement of motor skill itself, because infants usually are able to sit one way or another, but the quality of that sitting may be very different. So in some follow-up studies, Zuza has now another project still going on in our lab, centered on a longitudinal study, but centered on the acquisition of sitting. So looking at pre-sitting infants, freshly sitting, independently sitting infants, and expert independently sitting infants, so that we can look at developmental trajectory and transition in vocal production, in attention, around this major milestone achievement that is reorganizing everything in the motor system, basically. Yeah.
Hi, thank you. Very fascinating topics. Just a thought that just came to my mind is, I'm not an expert, first, sorry. But I mean, in some other mammal species, the newborns are already moving, so they are already quite highly developed. So motor development is quite ready. Could it be possible that for humans, this motor limitation is to help brain to develop more social interactions?
That's a very interesting proposition. Well, obviously, social interactions go on from the prenatal period. Vocal, there's vocal production. There are, movement responses. There are facial responses to stimuli. We know this from ultrasound, 4D scans, and there's increasing body of literature. Whether that is supposed to happen in the interest of social interactions, well, I'm not sure, because I guess for me more adaptive situation would be where you're kind of pop out, ready to go, right? Fully interact, engage, have more advanced motor skills. It's a very kind of tedious and difficult process of actually establishing social interactions with the newborn, right? For the new caregiver. And also, it's a very difficult process for the infant, interrupted by sleep all the time, and these huge fluctuations in wakefulness that disrupt your, social interactions all the time as well. So, I guess maybe the part we don't understand at that age is brain rhythms, specifically, of different stimulation. But I still think that the motor aspect is rather a biological constraint of how we're developing, because we see this in other domains. There's tons of literature from Karen Adolph talking about like how crawling completely shifts your worldview, because suddenly you can look up and you start to see other kinds of stimuli. How standing up completely, again, reorganizes your experience of the world. So far, the developmental literature seems to kind of show the opposite effect of motor, domain constraining rather than kind of enabling, early social interactions. Yeah. Okay.
Thank you so much. And other questions? Okay, so Przemek, thank you very much.
Thanks again.
It was excellent talk. We have a small gift for you.
Oh.
And you can notice that the mug will match your sweater.
Oh, that's so fantastic.
It was on purpose.
Thank you. That's lovely.
Thank you. Okay, so let's move on, because we are just good on time. So another talk, again, a group from Trento will talk about the ingredients of synchrony. So, yeah, the floor is yours again.
Okay, so for this second part, I will talk a bit more about what kind of aspects we need to take into account when we design an interpersonal synchrony study, and what kind of challenges especially we are going to face throughout the whole process. So I brought here a picture of a typical neural synchrony study, just to show how complex is the setup of the experiments. We typically have cameras, we have the dyad, in some cases one of the members of the dyad is a child. We have the neuroimaging part, or the physiological part, or whatever you like to use. In some cases with two devices, so we need to find ways to make these devices talk to each other. We have a task, which might be more or less structured, and basically all these aspects brings us gives us a lot of degree of freedoms in the choices of our experiments that we're gonna run. And so for today, I was thinking we could talk a bit about these aspects. So which type of dyad we're gonna enroll in our experiment, what kind of control we're gonna have for that dyad, which kind of task we're gonna have in our experiment, which kind of stimuli, in case we have stimuli. Otherwise, if we have a naturalistic interactions, which kind of stimuli are we looking for? Which kind of behaviors are we looking for throughout the interaction? Which modality we will look. So we will focus at the behavioral level, at the neural level, at the physiological level, all of them together, and more or less, and especially the lab setup, and ultimately, which is something that we have talked a lot throughout this workshop, how do we define synchrony for our study, how do we choose the metric, and how do we interpret synchrony for our task. So to start, what type of dyad do we choose? We have there is an article from Ruth Feldman, published in 2017, that gives us a theory of interpersonal synchrony at multiple levels. So hormonal, behavioral, and physiological neural level. And Ruth Feldman provided a very nice description of what we should expect synchrony to look like at each individual level for all these dyads. However, there are several challenges when we run a synchrony study. So, okay, we can choose one of these dyads, and the ones presented in these theories are parent-child, romantic partners, friends, and strangers, with synchrony decreasing in this order, theoretically. And we can design an experiment by having a within dyad experiment. So we have the same dyad repeating the same different tasks, so having different tasks, and basically comparing the same dyad throughout the whole experiment. This is a very powerful design because we have the dyad that is a control of them for themselves, right? So we can use we can have the same type of couple, which might be parent-child, involved in different types of experiments, and we can see how synchrony changes based on the difference, the aspect that differ among these tasks. However, if we want to compare, when I have a task, compare how different dyads, how is synchrony for the task across different dyads, we have to choose a between dyad experiment. And this is very interesting, but it poses several challenges because consider the fact that when we are in an hyperscanning studies, we're already talking about the double of participants that we need for a single participant study. And if we have a between dyad study, we need even more than that. So there is this aspect of the sample size that when we design an interpersonal synchrony study, we always need to take into account. And then ultimately, what do we use as a control group? Because we might have, as I said, different types of dyads, different types of relationships, and see how synchrony for the same task is differs among them. Of course, this is based on the assumption that we should find a difference between dyads because it's not a granted assumption, right? Many studies use strangers as a control group, but no study. I mean, the effect of strangers versus other types of dyad is not that clear. So we might need to choose other control groups. We might have a control task that is based on having a social situation, so two people in the same room, but we need to choose a non-social task, which is very, it's not that trivial to have. Another option, which is the most used, I would say, nowadays for synchrony studies, is to have the surrogate dyad group. So to have dyads that took part in the experiment, but that, as I said before, didn't interact with one another, so they are randomly matched from our database. And of course, I mean, this is still not the perfect solution, but I think it's the closest solution to the problem because these dyads took part in the same experiment, they did the same task, but they didn't interact. So we have a control of the direct social interaction effect. As I was saying, a challenge when comparing dyads is that despite the fact we have a strong theory that tells us what we should expect to see when we compare different relationships, we don't really have strong comparisons among types of dyads. So the theories would say that the closer the two participants and the higher the synchrony. But there is, for instance, a study conducted by Andrea a few years ago that shows the opposite. So strangers at the physiological level show higher synchrony as compared to friends and lovers. And again, how much structure do we put in the task? how do we choose the task? So we can go from, so the literature goes from very structured tasks, so two people co-attending to the same stimuli, so they don't have to do, they just have to pay attention to the same screen, they are in the, to the same stimulus, they are in the same room, they share the same environmental stimulation, but they don't have to interact with one another. So in this case, we have a lot of control on the exposure to the environment, but participants are not really interacting with one another. So it's it is a social situation, but it's passive interactions because they're not really interacting with one another. On the opposite side of the spectrum, we have free interactions. So the participants are free to interact as they prefer, typically with, I don't know, cooperative game, conversations. They can, they have naturalistic experiments where the parent plays with a child. But in this case, what do we look for? The interactions might be very different from one another, so we don't have a real comparison between the groups, right? And in fact, these are the two key dimensions of social interactions, if you think about it. So how close we are to the other person, and what kind of task we are doing with the other person. So there is the theory of interpersonal synchrony suggests that synchrony should increase at all levels, should increase the closer we are to the other person. So it should be stronger for parent-child as compared to strangers. And it should increase with the interactivity level, so with how actively involved we are in the social interactions. Of course, this model was derived by combining different studies. So there was no real comparison of these factors in the literature, and we don't know if this is really true. And of course, this poses a serious challenges to reach a theory of interpersonal synchrony, because as I was saying, to know what the role of synchrony is in social interactions, we need to understand the context in which that level of synchrony emerges from. So to tackle this problem, this challenge, we had a very recent study, which is now under review, and it was conducted in collaboration with the University of Vienna, with Stephanie Hoel's lab. And in this experiment, we compared, we used the two levels of social interaction, so the interactivity level, so we had 3 tasks that differ on the degree of interactivity. So we had a task that was passive exposure to the same video, so it was co-watching the same video without any interaction. We had a semi-controlled task where participants were playing a cooperative session of Jenga. So they were free, of course, to play, but they had to follow our rules. So they had to give each other turns, they had to take one break at a time, and so on. And then we had a third task, which was free interactions. So participants could really talk about what they preferred. And then all these 3 tasks were performed by 3 types of dyads. So we had close friend dyads, we had romantic partners, and we have mother-child dyads, with the child being 5 years old. And this was to test the models we saw before. And in this experiment, I forgot to add it, we used FNIR CEPA scanning, monitoring the inferior frontal gyrus and the temporoparietal junctions of the participants. So here, when we compared true versus surrogate dyads, we observed a statistical difference on all of the regions. So each of these regions, synchrony in each of these regions showed we had higher synchrony scores when in true dyads as compared to surrogate dyads. And then we did an analysis on by using all synchrony scores from all regions of interest altogether, here I plotted all data from each individual channel, but in the analysis we used the linear mixed models, so we took into account the random effects for same cluster, same region of interest, same participants, to model the individual variability. And what we observed is that the results are the opposite, as the theory would say. So mother-child dyads had lower synchrony than romantic partners and their friends. In the same way, the results are the opposite, as the theory would say, regarding social interactivity. So we see the highest amount of synchrony in the co-exposure, followed by the cooperative game and then the free interaction. And this was observed even when we conducted the analysis at the individual level for each region of interest, of course, corrected for multiple comparisons. So this is the result for the social closeness. And this is the result for the social interactivity level. Another aspect in the interpersonal synchrony studies is what do we look for? Because we said it's nice, we have a lot of naturalistic experiments. We have typically the video recordings of those experiments, but what kind of behaviors do we look for? And we typically have two types of ways to code these behaviors. So we have microcodings. We can see, for instance, the mutual gaze, the communicative gestures, the language. We can code language aspects of the interactions. And then we have macro coding, so aspects related to the whole interactions. And then we have to choose between which modality we can choose for our experiment. So here I just presented 3, which are the most common. We have others, as I was saying before. So we might be interested at brain level. In this case, do we use an EEG? Do we use an FNIRS? It depends. And in case we use an FNIRS, what are the regions we focus on? So typically, the literature uses the inferior frontal gyrus, the temporoparietal junction, or the prefrontal cortex. But of course, you have to, as we were seeing yesterday, you need to choose before, so you might miss some synchrony in areas that you didn't cover with your montage. You might be interested of synchrony at the physiological level, at the behavioral level, and you might be interested on all of them and to see how each individual level predict other types of synchrony. And to do so, you need to think about a way to make all these devices, such as neuroimaging technique, physiological recording instrument, talk to each other. So we need to have something you need to think about before running the experiments is how do all these devices communicate with each other? How do you synchronize the streams from all these devices? And one possible solution, of course, is not the only one that we are now using in our lab, is to have a LSL protocol. So to have all individual devices using a LSL protocol to stream their data to a common file. So at the end, what you will get is a file with all the streams that are synchronized with each other, possibly with triggers, so that you have a segmentation also of the experiment. And ultimately, this is something that emerged very frequently in the workshop. How do we define synchrony for our study? Are we interested in coherence? Are we interested in coordination? Are we interested in only in-phase or also anti-phase coherence? And of course, all these choices at the end are reflected. We need to be aware of them, because at the end they are reflected at the methodological level. So they should guide us in our analysis for our study. And regarding the synchrony, I wanted to present you today a video that we have worked on in the past years with our lab. And Okay. Not again. Okay, and then we will talk more about these aspects and how they reflect at the methodological level later with Andrea. So for now, thank you all.
Perfect, great timing. Yeah, great timing, great talk, and also very nice introduction to the first talk just after the break, which will be on synchrony in dance improvisation. So yeah, do we have any questions to Alessandro? Yeah.
Hi, I wanted to ask about this surprising result in the different types of tasks and different levels of synchrony. Is there any chance that maybe a self-control aspect is influencing these levels? Because in the task where people are watching the same thing on the screen, but they are supposed to not interact, maybe because they're controlling themselves not to have this natural interaction, maybe they have more higher synchrony because of that, because this need to communicate is like expressed on a different level, or maybe you have like other explanation of this. I'm very curious about that.
Yeah, I agree that that's totally possible, that the effort not to not to interact with one another is influencing the results, and this was particularly strong for the mother-child diets that we were testing. So we struggle a lot not to have the child looking at the mother, or the mother looking at the child while they were interacting. So I think that's a possible explanation. In my point of view, it's also possible that the fact that we have the same environmental stimulation for both participants, as well as social interactions, which might be not active, but it's still a social interaction, at the end, together, they bring us to observe the highest amount of synchrony. Because of course, at the brain level, we observe an effect of what the surrounding environment is, how the surrounding environment is stimulating her brain. while for the others, for the other tasks, I think the effort was more on the active social interactions. So, I mean, also the kind of stimulation that the two participants were exposed to was quite different, right? Because they have a perspective of one another. They need to think how to respond. They need to elaborate to receive the other person's information, need to process them, and then they need to respond appropriately. So I think there is a level of complexity that maybe like lowers the synchrony in that kind of task.
And other questions?
Yeah. So my question goes related in the same direction. For me, it's really difficult to think how would you compare mother-child dyads with adult dyads.
Yeah.
And how was there any control of the different developmental stages of the brain, or how the difference of hierarchy? For me, they are like so different that it's difficult for me to think about comparing them in the first place. And in the other direction, in another study related to the strangers versus the others, I thought maybe when you're in front of a stranger, you are more aware of them, you pay more attention to them, and you, I don't know, you are more alert somehow, and then that's why you come to a higher synchrony. I don't know.
Yeah, so I totally agree with you on both points. So the first one, the fact that I go back 1 second to the result. I think here the maturational aspects of the brain and the way in which the brain process, the brain rhythms develop throughout life has a strong impact on the results. So I think that's possibly what could explain this pattern, because of course we are using an adult child, we are comparing an adult child to adult diets. So yeah, I agree that might be an explanation. So it's not on the interpersonal closeness that affect the results, but it's more, we need to pay more maybe attention to the maturational aspects and how the brain rhythms changes throughout our lifespan. The second, can you repeat?
What was the strangers that maybe we are more aware and we have, we feel more attacked?
I agree with you. So I mean, my idea is that when we are involved in an interaction with a stranger, we of course need to pay more attention. We need to, I think there are also studies that they show that we mimic even more strangers in order to, let's say, have a,
be likable.
integrate, sorry?
Be likable.
Be likable, we need to, exactly, we need to pay more attention to what we are saying, to what we, the face expressions that we show, the way in which we display our affect to the strangers. So I think the effect is reversed for a synchrony in the literature. So it's, I agree with you.
If I may jump into this conversation, there are studies showing that while friends can converge, friends can have different opinions because they know each other and they can just share different views, opinions, they can act differently, and they will expect to be understood by a friend. So while strangers, they converge because they try to mimic, they what you said, they try to be likable and so on. So okay, so I think we're ready for a break. Thank you very much for this talk and conversation. According to the schedule, we have 15 minutes of a break, so enjoy your break. And also, I just want to focus attention of students. We have this box. You can leave your email addresses, and we will send you certificates, okay? If you need. Okay, enjoy your break.
Noi la mettiamo lì e poi la riduciamo a icona.
This is my great pleasure to invite Julian to give you a talk on interpersonal synergies in social interactions, from parent-child play to dance improvisation. Before we had a great introduction, you saw tango, so now it will be a continuation. And Julian is a team member of Professor Rączaszek Leonardi, who you had on Monday. So she led European-funded twinning grant, similar to ours. And background of Julian is computer sciences, but he joined, and we are very lucky, he joined Faculty of Psychology, and he does a lot of strong computational analysis, and he supports a lot of team. And he also conducts his own studies related to synchrony. Yeah? Okay, so the floor is yours.
That's right. Thank you very much for having me here. It's a great pleasure. Indeed, I would like to talk about a bunch of different studies concerning dynamics of movement coordination in interaction. From a particular angle, which I think is characteristic for our group. So just briefly, let me introduce the Human Interactivity and Language Lab, which I represent here. It's a group led by Joanna Rączaszek-Leonardi. I would say we are an interdisciplinary group of researchers that seriously believe in complexity and emergence. What do I mean by that? What we believe is that cognitive structures that we want to study are more likely to be emergent effect of multiple interacting processes from different timescales, rather than effects of easily to isolate factors that we can measure in very controlled experiments. This is why we subscribe more to naturalistic studies of interaction, and we try to make sense of the natural complexity of the world. You may see it here. This is the natural complexity of the physical world with continuous signals, complex organizations, biological processes of living organisms, and somehow we want to learn how it is possible that we get from this continuous physical world to the world of language, abstraction, and finally maybe symbol manipulation. So very formal, controlled systems which are characteristic for human cognition. I would like to introduce some vocabulary today, which may be inspiring, I hope so. So rather than talking about interpersonal synchrony, I would like to frame this talk using the concept of interpersonal synergy. So what's the difference? I mean, what's it's all about? So if, in a very simplistic manner, we could define synchrony as some form of matching of events or signals in time, interpersonal synergy is already a postulated mechanism through which such organization may occur. So we understand interpersonal synchrony as some form of distributed higher order control structure which spans two or more actors. So this is something which happens in between interacting people, which is characterized by a functional reduction of degrees of freedom of this compound system. We can imagine in a very simplistic manner that each synergy controls the variability of the system along a particular axis. So it preserves, it stabilizes certain relation. If this is the relation here, you can imagine that the variance along this axis is minimized, while the variance along the orthogonal axis is left uncontrolled. So synergies are always selective in what they control and what they do not control. So this is the first part. And the second, which is also a crucial element of a synergy, is reciprocal compensation. Okay, it's complex vocabulary again, but for a concept which I think is quite natural. So the concept here is that a synergy happens where both components are actively engaged in regulation. So if there is some disturbance, then both elements actively react to this disturbance. So we may have a very complex coordination between different actors, which is established in a not synergetic way. We could have, for example, a regulation just coming from one side. For instance, when there is one person imitating the movements of another person, then movements might be highly synchronous, but not synergetic at all. So the postulate here is stronger. And of course, synergies, they go beyond synchrony, because any pattern of organization can be regulated in a synergistic way. So any kind of coordination, not necessarily synchrony, but more complex patterns, can also be regulated by synergies. So this is the complex vocabulary. But in fact, what I want to talk about are what happens in natural interactions, something which we are all very familiar with, starting from parent-infant interactions through all sorts of social interactions, all to elaborate culturally selected forms of dancing. We make this assumption that all these activities are facilitated by interpersonal synergies happening on different levels, that this synergistic description is not limited just to movement coordination or simple behavioral coordination, but it naturally extends also to verbal exchanges. So we see a continuity between movement and language. And we make this assumption that those interpersonal synergies are the scaffolding which actually makes any form of social development possible. Why? Because through interpersonal synergies, degrees of freedom are reduced. So we can get from a very complex situation to something which is greatly simplified and manageable. Of course, this simplification is only contextual. It happens only in a single moment. So this is the conceptual framework. And I would like to go through a couple of different studies which illustrate different levels on which interpersonal synergies may occur. So starting from the very beginning of our sociality, interactions of mothers and infants. This is so-called diaper changing corpus. This is a joint work of Joanna Rączaszek-Leonardi with Katarina Rolfing and Iris Nomiku. And the data set basically consists of longitudinal data where diaper changing was recorded for the same pairs of mothers and infants throughout the basically first year of life. And the data was then manually coded so that occurrences of gaze for both mother and infant, moments where infant is gazing at the mother and vice versa, and occurrences of vocalization were coded in the videos. And they were analyzed using cross-recurrence analysis. And this is something which we will see in all of our studies. Basically, the method here is to look at the two time series. Here is the infant's time series, here is the mother's time series, and analyze separately all possible combinations of time points for the two time series. So if for a given pair of time points the same behavior occurs, so behaviors match, we make a dot here on the plot. And then we may, for instance, this is one way of quantifying it. There are different ways to quantify this plot. One way is to take the average over different diagonals of this matrix, which gives us something called a diagonal profile. And this is a rough measure of what's the level of coordination for the dyad at a given time delay. So you may see, for instance, leader-follower relations from that. Okay, so this is how the data was analyzed and what are the basic findings. So first, on the level of gaze, we observed that there is a particular interactive structure which emerges developmentally. So it's absent for the 3-month-old, but with infants of 8 months olds, we already see a very consistent synchrony when it comes to gaze. So they synchronize with their parents in the gaze behaviors. And of course, this is also a dyadic effect, right? It's not just the infants, but also mothers, presumably. But then when we look at vocalizations, something opposite happens. So we are already moving beyond synchrony. What we actually see is a separation. So we see that there is an active avoidance of simultaneous vocalizations. Rather, some basics which allow turn-taking already emerge. So this is the development which happens between 3 months old and 8 months old. Can we go further if we look at the types of vocalizations that are involved? So the data was coded according to some scheme and divided into speech-like and not speech-like vocalizations. And now it turned out that there is also a different pattern here. This is the mother following with her own vocalization. And it happens reliably for the speech-like vocalizations, but not for the non-speech-like vocalizations. So this is already a functional segregation of a vocal layer where some particular vocalizations become selected by the mother and they are met with a certain response. And the difference is only in timing. So there are no other events to support that other than timing. And presumably this is the structure which is necessary to scaffold the language acquisition. Okay, so this happens early on. Now we will jump a couple of years to the future, and we will have older children. So what's the continuous trajectory of social development? Now we have already toddlers, and they are playing with their parents. They are engaged in a particular joint action task. This is the task. Basically, it's a wooden labyrinth, and the task is to guide a ball through the labyrinth by pulling on the strings from both sides in a coordinated manner. So it's a quite complex joint action task, which we selected to have some context for interaction and to see whether there are particular preserved interaction patterns which potentially are associated with the children's social development. So here we are looking at between individual differences in the level of social development. Namely, we, let's say, calculated something like a theory of mind score based on the standardized false beliefs tasks. And we tried to observe whether this theory of mind score can be somehow predicted by the characteristics of motor coordination between parent and child. This is basically, those are coefficients of a linear model predicting theory of mind. Of course, there are some known differences depending on child's gender, working memories, as usually important for false beliefs tasks. But then we have a couple of different complexity measures which are taken from recurrence quantification analysis, which tell us something about the particular characteristics of a coupling between parent and child, which is associated with higher theory of mind scores. And what's interesting here, recurrence rate is the one which measures the overall level of coordination. And children with a high theory of mind score, actually, they display a lower overall coordination with their parents. So it's not a perfectly coordinated system. But what's important is that they follow their parents more reliably. So it's not about the overall coordination, but it's about functional coordination, which controls, so you can say it's interpersonal synergy, which binds the degrees of freedom that matter, while release the degrees of freedom which are not important for the task. And the selectivity and flexibility here is the key point. This was the study done with Arkadiusz Białek and Marta Białecka-Pikul from Jagiellonian University. So, moving further on, those two first studies were first about developmental differences, then between individual differences. But you could say that those differences are displayed on the developmental timescale. So we observed some coordination patterns here and now, and we assume that those patterns, they are somehow related to the general interactive scaffolding facilitating development. But now, changing the timescale, we ask a question whether we can learn something about the interaction happening here and now. And here we change the setting. So this is a study where we already have adults, although adults behaving in a childlike manner because they are dancing contact improvisation, which is a very childish activity in some ways. So they are having an improvised dance without pre-specified rules. Sometimes it's quite clumsy, but generally the idea is that they are trying to coordinate and be creative while communicating only through their movement, possibly physical contact. And we are asking this question whether by looking at the momentary level of movement coordination, moment by moment, we can predict how the relationship between dancers develop throughout this dance. So we had recordings of 7 improvised dance duets, and their level of togetherness between the dancers was annotated by dance experts, looking at the video recordings and annotating the togetherness moment by moment. So it's a continuous signal. And now we used a deep learning model to track the positions of individual body parts on the video recording. We actually analyzed the movement of hip center, so a center of mass of each person, and we quantified the coordination using cross-recurrence quantification analysis here. So this is the data for this study. Here, this is the standardized hip mobility for the 2 dancers. So you can say it's an input signal. Here from this, we calculate using a moving window coordination statistics through time. And the blue line is the average togetherness score as annotated by experts. And we are there's some pattern which is stable for all dyads, where first there's a warm-up when there's not so much togetherness and the coordination is not so large, and then it increases when they actually start dancing with each other. But what's more interesting is that when you look at the fluctuations, momentary fluctuations of the level of togetherness and the coordination statistics, for most of the dyads, you can see quite visible correlations between those 2 signals. And you could generalize it to the statement that maintaining a stable level of coordination, it yields a better interpersonal connection. In this context, of course, and it's context specific in this particular dance form, etc. And that stable level of coordination is a necessary condition, not sufficient, but necessary condition for forming interpersonal synergies. So our claim is that possibly what we observe is the formation of those interpersonal synergies within a given dance, within a given duet, which to the outside observer creates this impression of the greater togetherness between dancers. Okay, so altogether, we can have at least some evidence that movement coordination and patterns of movement coordination are important for understanding each other. But so far, we were discussing mostly nonverbal understanding and movement-related tasks. So how much can it translate to verbal exchanges in language? Because language, well, traditionally language is often understood as a carrier of abstract concepts. So something which is already a little bit disconnected from the immediate environment. It can be abstract, concepts like freedom or justice. Nobody have ever saw them physically, right? But somehow we use those concepts and we are able to talk about them. There are some theories which claim that, in fact, abstract meanings, even though they are not so much grounded in the physical reality, they are very much grounded in the social reality. So they're actually grounded in social interactions, and we need social interactions in order to understand abstraction and to develop abstract thinking, which simplifies a little bit to this statement that the more uncertain we are what some concept means, the more we need to rely on other person in order to define it, in order to understand it. And we believe that, again, interpersonal synergies would be what facilitates the process of meaning negotiation here. This is a study in collaboration with Anna Borghi and Chiara Fini from Rome, where we collected the recordings of 25 adult dyads, negotiating definitions of both concrete and abstract words over Zoom. And again, we used a quite similar setup. So we had the screen recordings from those Zoom conversations. We used a deep learning model to track the position of body parts. We registered shifts in the position of the tip of the nose of each interlocutor. So those were the time series, and we created recurrence plots from those, and then extracted different measures of coordination complexity from recurrence plots. And this is the main measure that we use. It's a measure of stability of coupling. And indeed, we observed that for abstract concepts, our dyads achieve more stable coordination than for concrete ones. So it's consistent with the theory which claims that understanding of abstraction is also embodied, and it's very much grounded in real-time dynamics. So what's the overall picture? Overall, I claim that we are trying to sketch this connection from the very basics, fundaments of embodied interactions, all the way to abstract thinking, claiming that everywhere along the line, the important processes are interactive processes. So we never decouple thinking from social interaction. We never become truly individualistic mind, but rather we are always in some complex web of relations with the others through interpersonal synergies, which provides the scaffolding for everything that happens. Thank you very much for the attention. We are sometimes organizing nice conferences in the mountains. It's a small advertisement. I'm happy to answer your questions.
This conference was introduced by me yesterday because this is my favorite concept of a walking conference. Hyperactive. This is definitely, yeah, we can see the question.
Hi, I wanted to ask about the study with creating definitions of abstract concepts. And I wanted to ask in which language participants were talking, because the dyads were from, what I understand, the people were from other countries, so they could either use their first language or they can either use English, and I'm curious which one you chose.
Mm-hmm. Yes, so all the people were talking in their native language. This data that I am showing you today is only from Polish participants. We wanted to collect also Italian dyads speaking in Italian, but for now we managed to finish data collection only for the Polish sample. So we are, yes, we are strongly requiring that this is the discussions are held in the first language. We don't have any very strong hypothesis regarding the between culture differences here, although we already know that the particular words are rated a little bit differently in terms of what kind of abstraction they make in Polish and Italian. So there are definitely language-related differences in understanding what is abstract and what's concrete.
Okay, thank you.
That's fine, I can walk.
Julian, thanks for an interesting talk. I actually wanted to ask you about including in some of your models dyadic measures from RQA rather than individual measures from RQA. It is a bit tricky, but have you actually done any analysis where that would make sense and explain the data better than individual determinism, for instance?
I guess here it's the most vivid. Yes. So okay, the general answer is that I am very curious myself, and I feel that even though we and many other groups are using RQA measures for quite some time for studying interpersonal interactions, we collectively still don't understand well how to use them. And there's a lot of questions here. Here specifically, we didn't use, so we used only cross RQA, we didn't use individual RQA, although some of those measures you might argue concern specifically parent or child. So if you talk about laminarity, it concerns the stability of only one participant of the interaction. So in some sense you have both here. And at least in this particular study, we observed that the measures that relates to child are more sensitive than the measures relating to parent. So presumably this is, yeah, because we are also interested in predicting some, let's say, individual social skills of a child.
Okay, but in a sense you don't have a pure measure of synergy.
Yeah.
That you're talking about, yeah?
That I don't have a pure measure of synergy.
Yeah.
Yeah, so with RQA we don't have a pure measures of synergy either way. So as far as I know, the only way to, the principled way to measure synergy would be to make a strong assumption what kind of relation is stabilized by a given synergy, and then calculate variance along this relation. And without this, we only have a proxy measures. Yeah.
So it will be our last question, because then we have moved to another presentation.
Okay, I have a question about your first study regarding the dancers. First of all, I was wondering if the dancers themselves knew each other before the study, or were they like partners, you know, dance partners in real life? Or my second question is that did you measure in any way their feeling of closeness after the dance? Because from my experience at least, I've always heard in my life that that dancing is a language itself. Mm-hmm. And I was wondering if that had any kind of correlation with their feelings.
Mm-hmm. Yes, so regarding the first question, it was mixed. So some people knew each other very well, some people were strangers who just met. Although we made these recordings during a dance festival, so everybody was participating in the same dance event, and in some sense it already created a sense of community. As for their first-person account of interpersonal closeness, so we used some questionnaire measures, but they all suffer from a ceiling effect. So basically, they all enjoyed their dance greatly, and they all feel very close after dancing. But we also have some qualitative interviews, and now we are trying to extract more from the interviews.
Okay, thank you very much. It was really great talk. We have also a small present for you, and I think again Mark tries to match your sweater. So
Thank you very
okay,
much.
thank you so much. Okay, so now we are moving back to the team from Trento. And now Andrea, Professor Bizego, is going to give you a talk. He's an engineer working on a synchrony. He develops a lot of methods to help us to understand and measure and analyze synchrony in better and more valid way. So yeah, the floor is yours.
Should be this.
Hello, everybody. Thanks for having me here. This is a pleasure. As Agnieszka's anticipated, my background is in bioengineering, but we can still be friends, I think. Yeah, so we will talk about pre-processing of FNIR signals while my stomach is still processing the pierogi yesterday. So this is meant to be a hopefully, introduction to what we can do with the signal that we collected yesterday. So some of you participated in the hands-on session. Some of you maybe just followed the presentation in the morning. Basically is my personal battle that I'm fighting with students, is that you need to know what to do with your data. You cannot outsource the thinking process of what to do with the data. And most importantly, you need to be aware of what to do with the data before you start the experiment, before you start to design the experiment. Because some metrics cannot be collected, cannot be computed if the experimental design isn't appropriately defined to be able to collect those measures. So I don't want to spend so much time on what is FNIRS. What I think is important from this slide is that, of course, it's very, very portable. It is expanding. Many, many studies are already adopting. What is important, I think, is that FNIRS signal is not a simple signal because it has many, many components. So what you collect when you collect FNIRS data is a combination of information from many, many different systems. I like the word system that we heard before. Not all of them, they are interdependent. Some are independent each other, so some are not. But basically, this signal contains a lot of information. And this is not always a good thing, okay? So now it will be very, very basic, very, very concrete topic. We will not hear about how to interpret synchrony, something like that. But what I want you to focus on now is what we are dealing with, okay? We are dealing with signals. And typically when we talk about synchrony, chronos from Greek is time, okay? So we are dealing with time, okay? It's not a simple measure, a questionnaire that you can take once during the experiment. It's a measure that you continuously take. And this measure is just a series of numbers. And in the end, the aim is to extract some metrics, some information from this signal. Why? Because you need to statistically test something, and it's very, very difficult to statistically compare signals. You typically statistically compare data points. So one subject, one data point, one diet, one data point, okay? One experimental unit, one number, okay? Instead, signals are very, very numbers, so we need to basically go from signals, from time to one number, okay? So this is the point of all the processing of signals and FNIR signals in particular. And this is also true for any type of data that is collected during time. Videos, for instance, but also physiological data, okay? Any type of data that has time as a supporting domain. So this slide is still for my PhD, when I tried to make my mind up on what I need to do. My PhD was on the analysis of signals from wearables devices. So basically, I realized that all the physiological signal processing pipeline that I was developing had these 3+1 different steps. And as an engineer, I like to categorize things, to define rules that help me to deal with the reality, probably. So I like to categorize all the signal processing steps that I was doing into 3 different domains, okay? The first domain that this is not shared between all researchers. This is my point of view, but I hope it will help you to understand. The first type of signal processing procedure that you will perform, I call them filtering and, again, pre-processing, because in some domain, pre-processing refers only to this step. So basically, you have signals with many, many different components, typically also with noise. And what you want to do is to remove all the components that you are not interested in, okay? And this basically will increase your signal-to-noise ratio. And by noise, I mean components that you are not interested in, okay? It doesn't mean that you are throwing away components that have no information, okay? But basically, if, for instance, here is an example on blood volume pulse data from wearable sensors. So these small oscillations are the heart pulse, okay? The long-term oscillation are probably movements or something like that. So we want to remove some component. We want to obtain this that only has the heart pulses, okay? The second step, is the extraction of the relevant information. Try to characterize what we observe visually in terms of numbers. So in case of heart rate variability analysis, don't know if you are familiar with, this means extracting the position of the heartbeats, okay, which is represented here. And the final step is, okay, now we want to give some, compute some metrics that can summarize the pattern that we observe in time into some numbers, okay? And depending on the domain, in heart rate variability, this can be just maybe the heart rate, okay? Or maybe the components in low, high frequency bands, whatever, okay? Before everything, the signal quality check. And this is becoming more and more important now that we are using devices that are more and more portable, of course, because you might have motion artifacts, not only motion artifacts, you might have simple disconnection of the data, of the sensor. You might have many other technical issues that basically affect the data. So along this presentation, we'll go step by step through all these blocks, focusing on FNIRS, okay? But these, I think, are steps that you will do, you will perform regardless of the type of signals, provided that is still a signal. So let's start with signal quality. I will try to use some studies that we did to show you some examples. So here we were focusing on the assessment of signal quality. Back 10, 15 years ago, this was the assessment of signal quality of FNIRS data was still done sometimes, not always, but it was still done manually. So here we compared 4 different raters. We provided them a set of segments of FNIRS signals, and we observed that basically, okay, we only have 4 examples, but 2 raters focused on maximizing the sensitivity, and 2 raters focused on maximizing the precision. So in one case, we had signals that were collected as good or bad. In all cases, signals were categorized as good or bad, but some raters only categorize signal with good quality, only signal with good quality, okay? But they categorize signals with bad quality, signals that were really with good quality, okay? And for other raters, were the opposite. So this is a problem in both cases because FNIRS data are quite expensive in terms of time and collection, but also equipment and so on. So you don't want to have to spend a lot of effort in collecting data from, let's say, 100 subjects, okay, and then having that signal with good quality are only 20, okay? So you don't want to be too much precise, too much restrictive on the signal quality. On the other hand, you don't want to use the signal with bad quality in your signal processing pipeline, because those signals could affect the outcomes of your analysis. So the ideal will be to have high sensitivity, high precision. There are some other methods that were used at that time. One was using some metrics that were computed on the signals and then applying some predefined thresholds, okay? And so if a signal was in the good range for all signal qualities, that it was qualified as a signal with a good quality. Otherwise, it was not, okay? But this was also not very good in terms of sensitivity, okay? It was good in terms of precision, but not sensitivity. So in this paper, we tried to approach this problem using machine learning and deep learning, which basically improved a little bit the situation, still not optimal. But this is to say, is a step that typically you want to do when you use data, and you need to decide how you want to do that. And depending on the method that you are using, you should expect higher or lower sensitivity, higher or lower precision. Another study that we tried to do, we compared the signals collected from one lab, collected from one team in Singapore, and signal collected from another team in Italy using the same experimental procedure. And we tried to classify the origin of the signals, okay? And in some cases, for some channels, a machine learning model was able to classify actually quite well the origin of the data, okay? This doesn't mean, this means that there are some differences in the collected data, okay? And these differences could be detected by the artificial intelligence model. This doesn't mean that those differences will affect the outcome, okay, of the analysis, okay? We didn't test this, but it's just to provide evidence that maybe differences in the version of the instrumentation, maybe differences in the experimental procedures, okay? Maybe differences in the way experimenters wear the cap. You know from yesterday that is not a trivial task. So you should be aware that, as is quite obvious in actually, but these differences could affect your data, and especially in case of SYNCC-IN project where we have data collected from many sites. We need the more you can define the more precise the guidelines on the experimental design and conduction, the better it is, so to reduce differences. One other remark is, yesterday in the hands-on session, we dealt with signal optimization, which is at the beginning of the data acquisition. It is, if you have good detected signal quality, this is definitely a good sign. It doesn't mean that the signal quality at the end of the experiment will be good. You might have disconnection, you might have subject movements, okay? So one thing is signal optimization, one thing is detecting the signal quality. And one good indicator, unfortunately only visual indicator of good signal quality, is the presence of the cardiac component into the FNIR signals. And actually, one of the indicators that we used here was an indicator that aimed at detecting this component, okay? Unfortunately, once you define number and then you define a threshold, this doesn't work well in all cases, and in fact, these thresholding methods didn't work very well, but the idea of this method was actually to detect this component into the signals. So visually, you can tell that some channels are better than other, and this, if you start your signal acquisition, is a good indication that you are trying to observe. Okay, this is just detecting the signal quality done. Now you need to decide what to do when you detect a signal with bad quality. I don't have answers. I don't want to give you the best recipe, because probably the best recipe will be defined in the paper that will be published tomorrow, okay? So just be aware that you will have to take some decisions, and it's better that you take this decision before. So some groups decide to reject data, some other groups decide not to reject, but still, when we say reject, we need to define what are we rejecting. Are we rejecting just the block that is affected by bad quality? Are we rejecting the channel? Are we rejecting the subject? Different solutions, okay? Each solution is trying to preserve some value, probably, okay? Still, you need to decide before, okay? Not after. For all these steps that we'll see today, try to have an idea before. Otherwise, after, you will be looking for you will be fishing for big fishes, and then you don't know where to go. So try, and the best thing, as I will remark at the end, if possible, of course, try to find a paper and stick to the processing pipeline that is proposed there, okay? Okay, so this is about signal quality. The filtering step basically aims at doing two different things. One is the removal of motion artifacts, and the other one is the removal of physiological components. So motion artifacts are some components that affect your signals that have some specific characteristics. They are time dependent. This means that they are not affecting the signal throughout the experiment. They are very localized, so only some specific portion of the signals have these artifacts, okay? And also, as the result from a paper that we are preparing suggests, they are also spatially localized. There are some regions that are more sensitive to motion artifacts, okay? This is a problem because you cannot apply a filter that processes all the signals at once, okay? You need to find a way to, because you don't want to alter the signal if you don't need to, right? You want to keep the signal as it is. So you want to just correct maybe the signals that are on the specific brain region, maybe only some specific portions. So temporally and spatially localized. A problem with motion artifact maybe doesn't affect interpersonal synchrony, but it affects, for instance, functional connectivity, is that if you have, let's say, a signal that has a correlation of 0.5 naturally, and you add the motion artifact, okay, if the motion artifact only affects one signal, then this correlation is reduced. But if the motion artifact affects both signals, the correlation is artificially, let's say, increased. So you might compute a correlation of 0.92, but this is only an effect of the motion artifact. So we did this study when we asked participants to perform different movements at different speeds and on the three main axes. So left, right, up, down, bend left, bend right. So we controlled the movements, and we were actually expecting that the magnitude of motion artifact was associated with intensity of the movement, right? So the faster you move, the higher the amplitude of your movement, the worse the motion artifacts. We found this is not the case, of course, only on 15 participants, but really these initial results really don't suggest there is a correspondence between intensity of the movement and motion artifacts, okay? Here, we computed two motion artifacts indicators. One is we tried to characterize the spike, so the range in a specific portion. So when you have a spike, that range increases, of course. And one other indicator of motion artifact is the baseline shift. So we measured the difference between the baseline signal before and after the movement. And we knew when the movement was performed because we controlled during the experiment. So these are still under review, but I think they provide clear indication that it's not really the intensity of the movement, but is the type of movement, and specifically some types of movement affect more some regions. So I go back here. The up-down movement affects more the occipital regions, okay? While the bend left, bend right movements affect more the left and right, let's say, part of the brain. And we interpret this as a result of probably very small shifts in the head, in the cap, in the occipital region, because this region is compressed when you do this movement, or it is stretched when you do this movement. Okay, so we don't know actually why, but we were expecting a correlation between motion artifacts and movement intensity, and we don't see this correlation. So yeah, don't expect that because your subjects are moving a lot that they are, you will find more motion artifact in the signal. This is not necessarily true. So when you want to avoid with motion artifacts, basically you have to perform two different tasks. The first is the detection. So when is your signal affected? Which portion of the signals are affected by motion artifacts? And the second one is the correction. So once you have identified motion artifact, then you want to try to recover the original signal, so the signal without motion artifacts. Unfortunately, none of these two steps can be informed by ground truth information. So all algorithms that have been so far developed, I will not say all because for sure there will be one paper that did not read, but definitely you don't know when your signal actually presents a motion artifact. You don't know how your correction algorithm is performing because you don't know the original signal because it's already altered by the motion artifact. Yeah, that is an issue. Of course, there are algorithms to deal with these two steps. I typically rely on the results of this paper that is for infant data, but I think can be ported to also adult data. And they say that, okay, they tried different pipelines, and they saw that the combination of two main algorithms, so the spline correction and wavelet filter, provided the best results. As I say, probably tomorrow a new algorithm will be published that will outperform this algorithm. You don't know, okay? But also, I decided to, personally, this is my personal choice. I'm not saying that you have to do the same, but I think it's also sometimes wrong to try to update your pipelines every time a new algorithm is delivered, because you lose the possibility of compare your result with previous results. So you need to balance, of course. This is an art, not something that you can really learn. One small remark that I want to do is that I foresee that in short term, deep learning will break this field of research as well. Deep learning means that basically you provide raw data to an algorithm, and this algorithm provides the result that you expect. We use deep learning in the study that I was mentioned before to automatically detect the intensity of the movement, because we used automatic video annotation of head pose. And so from head pose, so from the 3 angles of the heads, we could detect the movement, the speed of the movement. And we use this just to have a measure of movement intensity, okay? But definitely in future, there will be some group that will provide us with an algorithm to detect and correct motion artifact. The problem with deep learning, with development of these algorithms, is that they need annotated data. So they need a lot of annotated data. So I think if you want to collaborate, we can share the annotation, the data, and maybe be a group that maybe try to do something in this sense. Okay, I don't see many other group with this kind of synergy that can really provide this data and with the computational computer science skills to be able to do something. So I think we are in a good position if you want to do something in this sense. This is something we already saw. Probably this is the 3rd time now. So we know that our signal is not only the brain response, is also other components. And you already know about short channels, okay, which is a way of collecting information about physiological components. Okay, this is just a way to collect information. Then you need to decide what to do with this information. Will you use one short channels, all short channel? Will you average all short channel? Will you use short channel depending on the distance between the channel that you are focusing on and the short channel? There are many different approaches. So again, you need to decide in advance what is your approach. Another component is not using short channel, is just using signal processing. So basically the idea is that if physiological components are, the physiological components would affect all channels at the same time, more or less. So with principal component analysis, you try to extract the components that are shared between channels, and you will remove that component, more or less. But this is hard to do when you have small montages, because it's very difficult to discriminate what are the shared component because of the physiological component, or shared component because shared brain response. Okay. Another approach is using some other type of filter that basically remove the dependence of signal values from previous signal values. And this is particularly interesting and applicable when you approach FNIR's signal with GLM analysis. So again, maybe there are also other approaches. These one are the one that I kind of tried. I'm not recommending one or the other. This really depends on your needs, on what your experimental design, what your idea of experiment. So now I would like to, I think it's very educational, I don't know, to go back to the origins, I would say, of FNIRS. So FNIRS as, using FNIRS as to study brain response, because it was the original application that was somehow substitute of fMRI analysis. And after my next talk, we will talk about other approaches. So here, what I try to graphically represent is, this is the experimental design. So when you see one here, assume that your participant is looking at a stimulus, okay, very simply. And this is the expected brain response. So it means that if your brain region is responding to this stimulus, you would expect this type of signal pattern. You will expect to observe this type of signal pattern in FNIRS signal, okay? And this is called convolution, but you can think of it just like a simple multiplication. Okay, so you multiply the stimuli, okay, and the expected response for each stimulus, and then you have your expected signal, okay? Of course, there is this important coefficient, the beta coefficient, that tells you if the strength of your brain response, okay? So if beta is equal to -0.1, your expected brain response is this, where actually you see a decrease in the signal levels, okay? Small decrease because the absolute value of beta is small, 0.1, okay? And these three plots are on the same scale. Yeah, I can tell. So if beta is equal to 1, you expect this. If beta is equal to 2, you expect this, right? This is our expectation. This is our model, okay? And based on this model, now we analyze real FNIRS signals. The aim of this analysis is to extract beta. Remember at the beginning, sorry, just to recap, okay? I told you the aim of the signal processing is going from one signal to one indicator, okay? So this beta indicator is the number that we want to extract that basically tells us the strength of the brain response. It's one number, but is also affecting a signal, okay? So what we do is we subject by subject, we use this GLM approach where, okay, this is a bit complicated. Okay, we have time. On the right, you have your measured signal, okay? On the left, you have your expected signal, okay? This is just this plot rotated, okay? And beta, bold, is actually an array of values, and this one that you see as a figure is actually a matrix with numbers, okay? So in the first column of this matrix is the expected signal, okay? Then all these other components are used to filter frequency components from low frequency to high frequency components, okay? This is as an alternative to filtering, for instance, to use a bandpass filter. Can be, you may have 5 components, 10 components. This is the default design matrix when you use a Python library called nlearn, okay? So basically, what we want to do is to solve this equation where this is our output, and we want to explain this output as a combination of different input regressors, okay? Each one associated with its own coefficient. Is it clear? Time? Okay. 4 minutes? Is this clear, more or less? Okay. This afternoon, for those of you who will participate in the hands-on session, we will do it really manually, okay? But it's just a linear model, okay? Instead of having single number, you have signals, okay? like a cocktail, okay? You put together these signals, okay, and the final, the expected outcome comes out. And what you're interested in is the value of the beta associated with the stimulus, okay? After you have done this for all your regions, for all your stimuli, then you can compare between different conditions, you can compare different channels, okay? But the point is we went from signals, we filtered the signals, and then we extracted one indicator of the brain response. Of course, statistical analysis is not my field, so there are many, many different models that you can apply, multi-level models. So, but basically, it's just, at this point, you just have one table with one score for each participant and for each condition. Of course, as I anticipated, this is a fast developing field, so update your processing pipelines when it's needed. When it's needed, you tell me, okay? You see if the pipeline, the new algorithm is specific for the experimental design that you are using or you want to use, okay? I didn't mention the fact that each algorithm has its own parameters, and changing these parameters, you can change the result of the algorithm. So you not only have to choose the algorithm, but also the value of the parameters, which might expose you to overfitting. Okay, so typically an error that I do many, many times is that I try to create my pipeline and to adjust the parameters based on the data of one subject. Then I try, oh, let's see how this works on another subject. It is a mess, okay? So if you can try to avoid overfitting your pipeline on few cases, okay, and trust the fact that if there are if your pipeline is not really effective on one subject in general, okay, you can trust the fact that this goes in the variability of your data that will be managed in terms of statistical analysis. And try to avoid, if possible, subjective measures, okay? Try to avoid situations in which you have to decide based on signal, okay, whether or not to apply something. Try to decide it in advance, and if possible, use objective measures based on machine learning, based on algorithms that have been proposed previously. Yeah, this is a slide that I already showed in the kickoff meeting. I feel when I do this analysis that we are in this situation where we are not a clear idea of what we are doing, but still, this map probably was helpful for the next explorer that at the end of the travel, draw another map, maybe more precise map, and so on. So we are still in our process of getting to a kind of ideal exploration of the domain, okay? But I think we are still at this level. This I can skip if you want, because I will bring it up later. But basically, if you do adopt open science paradigms, you would avoid many, many problems when you process your signals. We'll talk about this later. So, thanks.
Thank you very much. I think we have time just for one question because we are a little bit behind the schedule. Andrea will join us again, so you can also keep your questions. Yeah? We are keeping the questions.
Thanks.
But I don't want to keep, I will to you. And again, a little bit different blue, but again, blue. So matching the mark, the color is matching your sweater.
Yeah, we agreed this before. So
Yeah.
thanks.
Thank you so much. Is it working? Yeah, and now we are moving to clinical psychology and to preterm babies with the talk of Professor Kmita from Faculty of Psychology, Warsaw University. Her background is clinical psychology and developmental psychology with the specific focus on preterm babies. She does incredible job on examining early social interactions between children, small children, and their parents. And it's my great pleasure to invite you, Grażynka, and thank you for accepting our invitation.
Thank you so much, and I'm really honored to be here. Thank you for having me. And now I think we'll switch a little bit to the person that we want to study. So if you are fine with that, I'll talk less about synchrony. I'll talk more about who is our participant. And as I understand, in your project, you plan to include preterm babies. Not babies, maybe. They would be offended, I think, if they are preschoolers. But anyway, so today I'd like to first focus on the preterm population and to look at it from more global towards a more nuanced view. And then I would like to talk a little bit about behavioral synchrony, but I think after Alessandro's lecture, we know all about it, and also the lectures that were on Monday and yesterday. So maybe I'll just briefly refer to that. And then the last point will be my thoughts about the challenges in measurement and actually bridging the gap between measurement and interpretation, because this is the biggest challenge. So first of all, I'd like to start with the scope of the phenomenon. So if you can see globally, around 15 million of babies are born before 37th gestational week, which is the definition of prematurity. And as you know, this is not, I would say, similar or unified group of children. We have different degrees of prematurity. And of course, I would say that the survival rates of these who are born very preterm or extremely preterm, very preterm is between 29th and 32nd week. And extremely preterm, these are children born before 28th gestational week. And this is the group that wins, in a way, having greater chances for survival, but at the same time, this is also the very group where most, let's say, most vulnerabilities might be met, which does not mean that I'm going to say that the rest of preemies, which are the majority, are without, let's say, all sorts of developmental challenges. And I'd like to emphasize that even though we are so, let's say, sophisticated in terms of technical equipment and ways of treating these children medically, prematurity remains one of the main causes of infantile death and all sorts of medical and developmental problems. Also, this is a real challenge to the family and to parents and siblings, and I would say that the experience of having your child in the neonatal intensive care unit is a huge challenge to the family. I'm trying not to use the word trauma. I used to use this word before. I think a lot of the experiences which are already there might have this traumatic meaning, but this is much more in it, and I'll try to explain it in details. Also, I would say that if we all agree that human development is embedded in relationships, then prematurity might provide the most challenging, let's say, phenomenon in terms of relationships and mental development. We have to remember that there are numerous complications of prematurity, and most people realize that CNS insult are a major cause of different developmental, detrimental developmental outcomes. But I think more and more research is focusing more on the cerebellum now in the case of preterm babies. As we know, at the last trimester of pregnancy is the time where cerebellum is developing really fast, and there are a lot of processes that are going on there. And also, I think Catherine Nimperopoulos is a neuropsychologist from Canada who is trying to longitudinally follow this group of children and track the, the different, let's say, insults to cerebellum. So this is what we learn more and more. But also quite new information. I think there's a nice study published last year from Sweden, where they point to a very important information relating necrotizing enterocolitis, which is one of the important complications of prematurity, with the risk of neurodevelopmental disorders in this population. And here is a graph from this study, and if you see, they compared typically performing preterm babies with those who had some abdominal surgery and with those who had medically treated NEC, which is necrotizing enterocolitis, and those who had surgical treatment of necrotizing enterocolitis. And you have to see that the risk for any of the neurodevelopmental disorders increased many times in the case of this last group, surgical let's say, treatment of necrotizing enterocolitis. And even with medical treatment, the risk was higher. And this is also a very important message, because now there's a hot topic, and I'll also refer to it, which is autism spectrum condition, as Julia has said, said on Monday, and beautifully discussed the topic. But I think if it comes to neurodevelopmental disorders, ASD is just one of the disorders. And we have to remember there are also other ones, like ADHD, developmental coordination disorder, a developmental language disorder, which are even more common in this population. So I think maybe it's a more neurodevelopmental spectrum rather than specifically only neurodevelopmental autism spectrum. So we have to remember, and also there is a very nice paper from the early 20s by Sato and colleagues, showing that in children up to the age of 5, children diagnosed with ASD, in 80% of them, at least one other neurodevelopmental disorder is also present. So the picture is quite complex, I would say. Another thing is that we, of course, are really interested in looking for predictors of preemie development. And this is a very nice analysis which uses a fancy analysis of networks. And if you look closely at the graph that I show here, what's quite strange is that the factor that is positively correlated with those infants' cognitive index in Bayley scales is maternal education. Of course, it does not explain too much of variance. It's 2.5%, but intraventricular hemorrhages of 3rd and 4th grade are a little bit below, and it's also something between 2% and 2.5%. We have also male sex, which is important in the case of preemies, because we know that the chances for survival are much lower for boys than girls. And also, I'll come back to it, the risk of, for instance, autism spectrum disorder is quite different. Okay, also we try to find some models that would link prematurity and all sorts of developmental and contextual factors with later development. I won't dwell on this picture a lot. This is an example of a model that we can try to, if we have time, maybe I'll just try to, if somebody has a question to it, we can go back to it if we have time for questions. If it comes to autism spectrum, I'd like to say that we have to distinguish between the 3 things: screening assessment, research assessment, and clinical assessment. And this is a very important issue. And if we look at preemies, let's say the screening tools overproduce the risk of ASD in this population. And I usually mean, what is meant here is MCHAT, which is, and probably you can guess why, and what Przemek was telling about the connections between interaction and development and motor functioning. I think this is one of the main reasons these children have a lot of, let's say, challenges in terms of all levels and developmental functions. Also, but still, even in clinical assessment, it's 6%, which is quite high. And of course, when you read all sorts of papers, you will find some people say it's 5-fold more, others would say 10 times more, etc. It depends on what statistics you consider in terms of full-term babies, but it also depends on what subset of preterm children you take. Then if you look at, let's say, specific clinical picture, it's quite different to what is described in papers on autism spectrum. And also, from my own clinical perspective, I would say that there is an issue of timing in interactions. That, for instance, a child who is a premature child may have a lot of behaviors that might serve to compensate for his vulnerabilities. So these are some kind of coping mechanisms that they may use to cope with changing environment. And I remember one boy who was not in line with his peers. It was a video recorded interaction at home, and other children were initiating a play, and this boy was just overwhelmed. They ran to another room, and this boy was doing what they were asking him to do, but it was not possible to meet in the same space and time. The only positive interaction of that boy was where they were seated in a kind of, I don't know, swing or something, and singing together. And they even sing with different voices, I mean changing voices. So that was incredible, how setting and context might shape your, let's say, how affordances can shape your possibilities to make contact with others. And then if you look at the ratio of boys to girls in preterm population, there is it's different in comparison to the general population. 2 boys to 1 girl, which is, I think, 4.5 to 1 is in the general population. That's the rate, I guess. And if you look at the links to gestational age at birth, it turns out that there is a correlation in the case of girls with the gestational age. So the smaller they are, the more risk for symptoms of ASD, whereas it's there is no such, I think in the case of boys, independently of the gestational age, they are at higher risk. And then there are multiple challenges which probably are due to what I've said at the beginning of my talk, this variety of let's say, physical and medical problems. Plus, I would add that parents of preterm babies are also facing more symptoms of PTSD, depression, and anxiety. So as partners for the interaction, they probably face bigger challenges. But mind you, again, typically in the studies, there is some point taken and we say, oh, mothers of preterms are at elevated risk of depression. But there are only few studies that try to follow the course of depressive anxiety and PTSD symptoms with time. One of the few I know is by Brunson from 2029, and it seems that it's quite important when you measure the symptoms, because it might be quite different depending on the month or whether the children are, for instance, at the age of 5 or 6, where studies show that there is not much difference, actually. Is it maybe a new brand, actually quite fresh paper from 2025, where there was a connection found between heart rate, let's say, abnormalities in preterm babies and the later risk of ASD. So that's interesting. That's just one paper. We need more research, more data on that. And of course, there are different mechanisms which are implicated or hypothesized and checked in research. Maybe let's go to that more, let's say closer look at the NICU experience, because as Stilwell and Harman said, there is a lived body and living body. And we have to also acknowledge that when we study anything in psychology, we have to reflect on the perspective from which we study our participants and the phenomenon we are interested in. And following Buber and Vasudevi Reddy, that was already mentioned here in Przemek's lecture, I would say that a lot of things would not be discovered unless we assume a second-person perspective, which is actually being in interaction with the other person. And if you look at the NICU, and you always have to see, if you have findings of a study, you have to really ask the authors to describe the context, because NICUs are different. They are, I haven't, Agnieszka, do I have some time still? Yeah, more or less, okay. Yeah. No, not so good, not so bad. Yeah, okay. So if you look at NICUs everywhere, there might be quite different context. For instance, whether mothers and fathers are allowed, for how long, what parents can do with their children. Can they kangaroo if a child is still mechanically ventilated or not? How pain is, let's say, managed in a unit. And many other. How can you talk to doctors and nurses and midwives? Who's responsible for what? What about the decisions? So that all matters, I would say. Also, if we look at behavioral data, we scan this or that, it's very often the third person perspective, but perhaps our one of the good ideas would be to couple the different perspectives. So I try to do it in a mixed method studies where for instance, in our old study of interactions of preterm babies from the first month of life corrected age till the 12 months, we always, after recording interactions, we were talking to parents and asking about what they felt, how they compare the situation to the real life situations, whether there was something that was astonishing to the parents, etc., etc. I think if you combine, it can be also interacting with a baby might change your mind on what is possible to see. And here, as you can see, these parents are experiencing something which is very close to traumatic experience, or even is a traumatic experience. They experience complete, let's say, helplessness sometimes. They experience total anxiety about the life and health of our child. But at the same time, in a small moments of meetings, they meet a real person who holds daddy's, hand, or who opens their eye, or does a lot of things that are meaningful in this second person approach. So I think this is quite different if we compare this more ecologically valid and closer, if we draw our attention closer to what's going on in real life. Also, in terms of pain, this is what we simply counted, painful procedures for our most vulnerable infants born below 26th gestational week. And the last column shows you number of stressful procedures / number of skin-breaking procedures. And if you look at the girl who was born at 22 weeks and I think 3 days with 500 grams, and she stayed with us for 147 days, her number of skin-breaking procedures throughout this time was 354. That's unbelievable. And if you look at the brilliant set of studies by Montirosso and Provenzi, and you look for the connection between neonatal pain due to skin-breaking procedures and then what's going on in terms of temperament, attention, stress reactivity in terms of temperament, then emotional reactivity, and then socioaffective quotients, you see that via the processes of gene methylation due to these early experiences, there might be a reprogramming of temperamental, let's say, profiles of these babies with all sorts of long-term effects for their later life. This slide was actually taken from a presentation of Livio Provenzi that was conducted virtually last year here in Warsaw. And also, maybe I'll skip this, but I also wanted to say that even doing study in the NICU, which seems to be a one-time point, we have to be really careful to where we are in the process from this, I don't know, fragile baby that needs to be cured, protected, and monitored all the time, to the baby with a social sense, who is an interaction partner and who is an active participant in those moments of meetings. So I think it's also a very important part. Christine Neugebauer, in a paper from 2022, she suggested that, because everybody agrees that the quality of early interactions are super important in the case of vulnerable groups like preterm babies. So, but how do we assess it and how do we conceptualize it? And here is one of the models where these all different levels of interpersonal dynamics might be and what factors should be considered. So if we come back to the myriad of terms that we discussed during those 3 days, we have dyadic synchrony, we have all sorts of synchrony, like behavioral, it means dyadic, bio-behavioral, which is already a kind of a synthesis, trying to capture the different levels of the interpersonal processes. But we also have co-regulation, coordination, mutual regulation, interactional regulation, attunement, reciprocity, as you said, convergence? Synergy, yes, synergy, wonderful. So you see, and these terms are close to each other, but maybe they are capturing different views and different levels of analysis. So for me, I really like the concept of interactional regulation because it's neutral, it's quite flexible. Actually, it was introduced by Beatrice Beebe, and you remember the famous study that was mentioned by Jean Nguyen, if I'm correct with the name, yesterday by Joseph Jaffe, Frank Lachman, and Beatrice Beebe on this mid-range coordination of vocal signals, which is probably optimal for development. But so today I'll focus a bit on interactional regulation, but also on bio-behavioral synchrony, because I think if we are close to the definition that was given here many times and to the model suggested and developed by Ruth Feldman and her co-workers, which actually is rooted in ethology, in phenomenology as well, and evolutionary psychology. I think maybe we capture something very important, like which is going on different levels. Okay, so now maybe I'll skip what is this bio-behavioral synchrony, why it is important. One of the latest papers by Ruth Feldman in World Psychiatry discusses the concept against another concept which is quite a hot concept for some time, resilience. And I think if we talk about resilience, it's a very important term in case of vulnerable populations. And if we compare interactions of preterm and full-term diets, I would suggest you reading a new systematic review by Hartzell and colleagues, which is a very nice paper. They have analyzed studies that were published between 1980 and 2022, which is a great job. They identified 190 studies and 52 met the criteria for the inclusion in the analysis. But if you look closely, there were studies that focused on maternal behavior, on infants' behavior, but also on dyadic specific behavior. Only 14 studies, and only 8 of them found less synchrony in preterm dyads. The remainder found no difference. So it's interesting that actually the data and the results are quite mixed. We have studies that support this idea that there is less synchrony and the quality of interactions is, let's say, worse in this group, but there are also studies that say it's better. That's wonderful. And there are studies, we can't find any change, differences. Of course, this is a piece from our study from the early, 2010, let's say, and this is a visualization of affect in triads. It was captured at the age of 12 months, corrected age for the baby, and mom and dad were present in the room. They were playing in the presence of a stranger coming to the room. And if you look at the full-term boy, you can see some kind of variety of different affective displays in the three participants. In another boy who was a preterm boy, born at 28 gestational week, it's not so rich. But can we say that the upper picture is better or more optimal? Or maybe they track different phenomena. Okay, so just the last word. This is our study. This is the study done by Anna Grochowska with my help on autonomic synchrony in mother-infant interactions and affective touch. And this part of a study, it was a prospective study on a small sample of 16 dyads, but it was also due to the fact that it was conducted during COVID-19 pandemic, and the NICUs were more restrictive than before, and the NICU we conducted the study was even, I would say, a little bit more flexible, but still, and we plan to include fathers, as I always include fathers in my studies. But the situation was such that we decided to go the easiest way. And if you look at this, we actually later collected data at 3 and 6 months corrected age, but I'll just show you a brief information from this first collection, which was data collection, which was at the end of infant stay, and the infants were without major, neurologic or other health complications. But what turned out, we tried to, first of all, we compared, we used natural experiment, because in this NICU, what was going on, that mothers could interact with our babies as they would like to do. And then at certain point, there was a time for blood collection or vaccination of a baby, which was a natural, maybe not nice, but natural procedure in this world. And after that, The mothers were expected to calm the baby, holding the baby, doing kangaroo care. And what we did was to measure HRV signals from the baby and mom with NIPE and ANI systems, and then to also annotate affective touch the mother was using to comfort her baby after the painful procedure. And what was interesting that there was more, let's say, synchrony in the free situation, but the entropy was higher in the free situation, which is, I think, quite a understandable result, because interactions are not stimulus-response situations. They are actually based on contingencies and on freedom to choose your next step. And I think this free situation was richer, more richer in terms of time series. And the quality of synchrony in this pre-pain procedure was correlated with the length of affective touch after painful procedure. My last sentence. But the tricky thing is that when we analyzed the signals of non-optimal interactive behavior with the use of attachment during stress scale by Massey and Campbell, it turned out that longer affective touch was also correlated with more maternal, let's say, either intrusive or not so active signals. There are different signals taken into account here. So sometimes I think it's difficult, but maybe what came to my mind is that affective touch is also not the good in itself. It doesn't mean the more touch, the better. Maybe this mid-range, let's say, phenomenon is also here. Okay, so I don't have time for more. So just I think we all believe in enacted science. I think it was I could hear it in your talk, Julian. And so I think there are now, so I would only say I'm very much forward to the 4 E's and even to 5 E's, which adds emotional, because I think it's all done in interactions are inherently emotional. And maybe that will be my last slide. So the biggest challenge is how to make sense of the different levels of interpersonal dynamics. And I wish you great success in it, in your project. And I refer here to the slide that was shown at least, I counted 3 times during this workshop. And thank you so much for your attention and for taking too much of your time. Thank you.
Thank you so much. So interesting talk. I'm sure we have many questions. I think we have time maybe for 2. And I just wanted to mention that we have a team member from Milan. She's a pediatrician working with the baby you mentioned. So sorry to put you into spotlight, but you might want to talk during the lunch break. So do you have questions? Sorry to be like, bad policeman. It's not a fair role, but someone has to.
Thank you. This was very, very wonderful talk. Really, I learned a lot. And I was wondering, because I concentrate on the parents, and is there any research that shows that having an autistic parent increases the risk of being premature?
To tell you the truth, I don't know about any paper like this, but I can, if you like, I can also do a search on it, because if you ask this question now, it stays in my mind, and I won't sit until I find it. If you can email me, and if I find something, I'll definitely help you. I can talk to my colleagues also from the faculty. Maybe Professor Pisula would learn more, because I think, but the reasons for prematurity are really complex, I think. And as far as we know, I think we know less than we don't know much, actually. I mean, there are some treatable reasons, but far more are just to be discovered, I guess.
Thank you. I will contact you.
Thank you.
And it would be also great to collaborate with you, Grażynka, on our project, because as you know, we plan to include this population, and so yeah, we would like to take advantage of your expertise. So, okay, so
thank you so much.
Gift
I'll be honored, and I tell you that that's actually, you realize my dream, because I was always looking at two different corners. I think your lecture was great for me also, but now I think something really important happens. Okay.
No, don't run away. A present for you. Thank you so much. Yeah, it was really interesting. Okay, I think you lost your scarf over there, you know?
Okay, I was using my hands so much that I lost my scarf. Okay.
Okay, so now we have time for the last lecture, again by Andrea, and we will dive into the analysis again, and then there will be a lunch break. So, yeah, Andrea, the floor is yours again. Yeah, you will manage, yeah?
Yeah.
Okay. Glad to see you again. So this second part is, would like to focus more on the computation of interpersonal synchrony. I will not give any answer as before, just maybe pose more questions. So just to recap, we know this, okay? So we need to go from a signal to a number. And for, let's say, I would say simple approaches, so where we want to measure brain response, we use this model, okay? And we basically compute one number that tells us the strength of the association between the stimulus and the brain response, okay? Easy to say, and probably also to do. But the point is, that we have this model. It makes sense, right? It was also derived from fMRI, which is probably older than fNIRS. So we have these models that we know how to do, okay? We know what is the meaning of beta, okay? Then, of course, we can interpret the meaning of the results according to the domain, the experimental design, so on and so forth. We still are at the beginning of our journey into interpersonal synchrony. So now what happens when we go from this type of experiment, okay, to interpersonal studies, hyperscanning studies, but also observational studies, whatever, okay. So the first thing that I realized that is much more difficult, because basically your experimental unit becomes the diet, okay? And so you need to double the number of subjects. This seems quite trivial, maybe don't need to mention, but it actually affects the effort that you need to put into collecting data. What we saw about the signal processing pipeline is probably, I would say, applicable to interpersonal studies. Apart that for some algorithms may be more appropriate than other when you are going to use to analyze data to compute some kind of synchrony between these signals. The real problem is how we can substitute the betas with another number. Okay, how do we compute a number that quantifies the level of synchrony between the two persons? And typically, you could use GLM designs, so basically experimental design where we have a sequence of stimuli, but as we realized many times during this workshop, we could, if we have, but we would like to target naturalistic studies because it's in naturalistic studies that you might observe more easily the social interaction. Although something that Alessandro said, I would like to maybe not correct, but he said, okay, in the passive view, okay, there is no interaction. I don't think this is true. We saw in the video, right, where the mother was redirecting the attention. So interaction is always there, okay, so, but still, GLM designs are typically less used when you want to study interpersonal synchrony. So let's try to, as I like to do, to define some general rules and constraints just to restrict maybe and to be able to focus more on what we will talk about later. So we focus on dyads. Of course, there are some studies that focus on groups, and that, of course, is even more complicated. I would say exponentially more complicated. In mind, we have non-symmetrical dyads. So when these terminologies, I made it. These slides are actually more notes that I wrote during my thinking about the problem. So these are not probably shared between the domain. But so when I say non-symmetric, I want to remark the fact that in, for instance, parent-child dyads, the role of one member is different from the role of the other member. So we might be interested also in the directionality and the meaning, the interpretation that we can give to this direction might be different. Okay, this one thing is to say that the father affects the child. One thing is to say that the child affects the father or the mother or whatever. I feel very involved, so I will say father many times. Another thing is, of course, we want to move to naturalistic design, but we should try to have a semi-structured design. So basically, trying to give a definition of what is a semi-structured design. Basically, you don't have any control as an experimenter to the order or onset, duration of the different tasks, okay? For instance, interaction, vocalization, movement, dance, whatever. But you can control that they are present, okay? And then information about duration, order, onset of the task can be derived after, for instance, using video recordings of the experiment. So you are able to categorize your experimental data according to different, you can able to segment, to portion different the data, so you are able to say, oh, here they were doing this, here they were doing that. And we've mainly referred to design where the same type of signal is collected from both members. So we have the same type of information from both dyad members. Okay, again, this I will stress you a lot about this today. So what do you think about when you think about biobehavioral synchrony? This slide is not new. Still, I used this during the kickoff meeting, but I think it's important because when you think, I think it's also important for us to use correct terminology. Synchrony can be that synchrony during diving, synchrony during dance. This is also synchrony, where different people do different tasks, and what one person does affects what the other person does, and to work, they have been synchronized, but they don't need to do the same thing at the same time, of course. So this is more behavioral, probably. Of course, behaviors affects and can be studied in terms of physiological responses, neural responses. And another thing is that the problem is that at a certain point we need to, once we identified what we will need to study, what we want to observe, we need a mathematical formula that allows us to characterize this, okay? So this is the problem that we will discuss today. So again, just a summary, there are many, many different possible level sources of synchrony. This is a paper that summarizes basically the type of synchrony that can be derived from the environment, from sensors, so sensory inputs, whatever. I appreciate that thinking about this, the problem, I kind of came to the same domain of the previous speaker. So I would say that if we want to talk about synchrony, we can consider the fact that synchrony is some kind of reduction in the degrees of freedom. So I will explain this. If you think that an F-near signal is just a random signal, okay? So a signal that can take any values at any time point. Of course, there will be some internal structure because F-near signal will have some autocorrelation. But in the end, can be the measurement of a random process. Of course, there are some factors that can limit the possible values that this signal can take, right? So one limitation can come from the context, from the environment, okay? So for instance, is the room temperature too hot, too cold, okay? Maybe your heart will react differently, okay? Or maybe because of the physical characteristics of the experimental setup, so the physiological demand. So this type of factors can basically reduce the degrees of freedom of your signal. So your signal will not be a random signal, but will be a signal that will be affected, will be influenced by these factors. Another possible source of constraint in terms of degrees of freedom of the signal can be that your brain is processing information, is trying to elaborate information, and these processes, of course, will affect the degree of freedom of your signal. In fact, we can say this, right? So our signal is not a random process, but is a signal that is expected to have some specific patterns, okay? As an effect of cognitive processes. And there are social effects, of course. So these are the effects that we are more interested in. And that means that these social effects can also have different sub-sources. Simply the fact that we are here together, this might influence, even though we don't know each other all, okay? But the mere presence, simply the fact that we are here, this can affect and reduce or maybe increase the degrees of freedom of F-near signals. Or maybe social stimuli, so people that smile, people that are angry, people that talk, okay? Whatever, I don't know, I'm not an expert actually in social neuroscience. So, but definitely these, I think, are the factors that we are more interested in when we talk about interpersonal synchrony. The important thing is that I think we need to, sooner or later, I think, try to reflect on the effects that each type of factor can have on our signal, okay? By the way, I was also thinking before, during the presentation of, inspired by the presentation before me, this can be true also in terms of behavior, right? So being in a social context would reduce our degrees of freedom, luckily. So we are not really free to do everything, but depending on the context, what we want to do, what we can do will be different. So let's say, again, this is just some notes, how I can try to order my ideas when I think about interpersonal synchrony. And I think that all these factors can affect both members of the dyads simultaneously, okay? So the point is being able to discriminate if what we observe when we measure interpersonal synchrony is due to the context, to the cognitive processes, to social affects. So being able to assign their actual numbers, so the actual quantify the contribution of the individual effects. I think it's very important this time to think about what are you studying, what are the factors that you want to focus on, and how you expect those factors will affect the degrees of freedom, the patterns in the signals. It's very important because, as I said at the beginning, the hardest part now is trying to identify a mathematical formula, a computational process that transforms your signals into a number that quantifies something, okay? And this something can be described, okay, using many, many different mathematical formulas. So starting from brain functionality, okay, we need to think, I think, in terms of, what happens to our brains, okay? And since, based on that, what we expect to observe in terms of signals, and what we expect, what we want, how do we want to characterize those patterns in terms of mathematical definition. I think this is a process that we should try to do every time we talk about synchrony. And after we do that, maybe we want to change the term synchrony with the actual metric that we are measuring, okay, the actual phenomenon that we want to measure. So just an example.
Blacktip reef
we have
sharks.
audio here. Well, let me just, okay. So I'm quite fascinated by these pictures, okay, where there are some sharks hunting and fishes that, of course, escape. I found a nice sequence before. Okay, maybe I just try to stop. Okay, so are they synchronized? Yes. Okay, the fishes are running away. Yes, they are synchronized, but how do you measure this? Of course, we can try to put a sensor on each fish and try to measure some kind of similarity. So, but you see, I mean, this is the process that we need to do. I don't say this is technically feasible, but we need to go from our concept of synchrony and try to give a concrete name, a concrete measure, okay? Actually, when I thought about this example, I was thinking about other example when this happened with humans, and unfortunately, when you have a big crowd that gets panics, okay, you may also have casualties. So you don't want this type of synchrony in humans, I think. Okay. Another type of synchrony, and here I'm borrowing from the study of Gianluca, study of 2013. So basically, now I will really go.
Mammalian infants are born immature, and
so across species, okay, we have some shared behaviors, okay, parent-child interaction, let's say. And what really intrigues me about this, of course, if you go this is Gianluca when he was young. So this, right? Is it going on? There is some. Oh, sorry. I know there is a way of also recording the starting point.
Heart rate of the baby during the task.
Okay.
The lower the heart rate of the baby while
we will hear Gianluca after. Okay. You see, the heart rate of the child decreases when the mother is carrying the child, okay? Sorry, I wanted to pause the video actually. Would you say that this is synchrony? I would say this is synchrony. Did you need to measure the heart rate from both the mother and the child to do that? No, okay? So this type of synchrony is, of course, is synchrony. We can say this is synchrony, but it's not the same synchrony that we saw in studies where we measure F-needs from both the mother and the child. You see my point. So you need to really try, I think. I will try myself to try to avoid the term synchrony and try to specify what I'm actually talking about. And in this video, I also very, very fascinated by this.
So the pup?
That basically puppets, they, this is, okay, normal and sensory deprived, but let's stick on the normal. When they are picked like this, they stop moving to facilitate the caring of the mother. Okay, very facilitated. This happens across many different mammal species. It happens with children, and this is an explanation how behavior affects the other behavior. But same, it's not that they are doing the same thing. Actually, they are doing the opposite, because if the puppy moves, the mother will probably leave it there because it's too difficult to carry the puppy if it is moving, okay? So again, two different contexts, okay? Okay, I'm messing around with these videos. I wanted to go to the previous slide. Two different types of, let's say, synchrony, but we need to specify what we are talking about. So what we are talking about when we talk about synchrony using functional near infrared spectroscopy, this you already saw, this graph on the left you already saw. Many studies that focus on interpersonal synchrony using F-needs use coherence. Coherence is basically a measure of similarity, okay? More complex than that, okay? Correlation is also probably a measure of similarity. Regression is also a measure of similarity. Is similarity what you expect? Is it always similarity what you expect when you study mother-child interactions? I'm not sure about that, okay? However, still we consider 4 similarity metrics, okay? And we tried to see if this was before the video that Alessandro showed you before. But basically, in this experiment, we had, I think, yeah, it's mother and father listen to different stimuli. And we compared the data when they were hearing the same stimulus and the data when they were listening to 2 different stimuli. Okay, so we randomized, let's say, our control condition was that the 2 members were listening to different stimuli. And we could assume that we expect higher synchrony, higher similarity, okay, when they were hearing the same stimulus. So we could make this assumption because experimentally we could control this, and we compare different synchrony metrics that at that time were the most used metrics. Not all metrics behave the same. Of course, they measure different things. And what most important is that not all metrics would pass a statistical test that tell that the 2 conditions were different. Okay. And again, I'm not saying that any of these 4 metrics is the best metric. It depends on what you want to measure. But you need to think first what you want to measure. Another thing that Alessandro was mentioning before is this framework through which we can have some kind of control condition. Basically, the idea is that we compute the synchrony metric between true and surrogate dyads. So true dyads are the actual dyads, our experimental dyads. Surrogate dyads are dyads that are created at processing time by pairing together a member of the dyad and a member of another dyad, okay, which did the same experiment. Of course, they were not doing the same experiment. And this is interesting because in case of experimental design where you have a stimulus, you could say that the synchrony that you compute on surrogate dyad is the synchrony that is due to the stimulus, okay? Because what these two members had in common is just that they were looking at the same stimulus, but at different times, they were not present in the same room together. And while the synchrony that you compute on true dyads is the synchrony that is both given by the task and by the fact that they were actually there together, okay? This is another process that you could do. So you could try to create surrogate signal that have some, that share some characteristics with the true signals and compute the surrogate, the synchrony between surrogate signals just to have a baseline level. So what will be your expected level of synchrony in case of artificially created signals that have some characteristic with the original signals? So yeah, there are many metrics, of course, but it's very important that you think before the metric that you want to use, and more important that you think what you want to study, and that will inform you about what is the metric that you want to use. Of course, there is not much expertise on it. I think this is one of the fields that requires more efforts in the future years. So now, going semi-structured, so basically moving from task-based or semi-structured experiment to naturalistic designs. We already discussed more or less the differences. The point is, okay, how do you compute synchrony? When do you compute synchrony? What are the portion of signal that you use when you compute synchrony in naturalistic design studies? This is an attempt we made. So basically, we annotated the experiment. So looking at the video recording, and in this case, we annotated joint, non-joint sections. So sections in the experiment when the two members were doing the same thing, or maybe, well, looking at each other, so some kind of joint task, okay, and section where they did not. Of course, we had different dyads with different experimental timelines because this was not controlled. And the problem is how do you compute synchrony, okay, because each experiment is different from the other. So one idea was, in our case, again, I'm not saying this is the optimal solution, but one of the possible solutions would be to fix a fixed length period, for instance, 15 seconds, and take the first 15 seconds of each instance, and then maybe take the average. So compute the synchrony across each portion, then take the average, okay? Unfortunately, as you could expect, the longer the length that you want to consider, the lower the number of dyads that actually have data that you could use. And this decreases exponentially, unfortunately. So again, naturalisticness is nice, but apart from signal quality issue, which might be more critical, but you need also to think about how you want to manage this naturalisticness in terms of computation of synchrony. So just to summarize, okay, I think there are three main issues. The first is defining the synchrony metric, okay? And this should be informed by what you want to investigate. And this should force you to think about what are your expectations in terms of the phenomenon that you want to observe. The second point is that synchrony is just one step in a signal processing procedure that also has some degrees of freedom itself. So, and up and downstream steps might affect what you measure, okay? Finally, you need some, sooner or later, to compute some number and apply some statistical framework. You need to define what are your design, if you have case control, if you use through or surrogate bias, okay? The important thing is do this before you start the data collection, okay? To avoid bias, of course, and to ensure data quality. And nobody will help you because nobody knows what is the true, not only in terms of research question, but also in terms of the appropriate signal processing pipeline. So it's very, very important that you rely on open science, okay? You share your data because that could help other researchers to improve the pipelines, okay? And sharing data is not only sharing the raw data, but also the metadata, so the data of the data, okay? So that tell other researchers how those data have been collected and how to interpret the data, and provide, if possible, contextual data, okay? Release your software, if possible, because that, of course, ensures the replicability of your studies, but also you can always go back and try to maybe improve the exact same data because you can compare the one signal processing pipeline with the other, and try to avoid subjective evaluation on the signal quality, for instance. And I think what most important is share also your knowledge. So share your good practices, share also your bad experiences with data processing, of course. Pre-register the studies. You know, there are two levels. One is just pre-registration, that is, say, just something that a record of what you want to do. One is the register report, which is more difficult, but I think guarantees the quality of the scientific results. Yeah, so I think we really are in the explorative phase, so we really need the help of everybody to advance interpersonal synchrony. I will not use synchrony anymore, possibly, so let's see. Okay, thank you.
Thank you very much. Do we have questions?
Thank you. I really like your conclusion not to use the word synchrony. But I wanted to ask, maybe are you aware of any comparisons of difference, correlation, coherence, mutual information metrics, different measures of different forms of organization, basically, between two time series, which try to give some interpretation to them in the context of interpersonal interactions, not in the context of physical signals?
So for the first question about the comparison, we try to do something, of course, we in a specific domain and with a specific experimental design, and we mainly focus on signals that were all about, all trying to measure signal similarity somehow, okay? I'm not aware of any studies that tell you that depending on the type of synchrony metric, the type of signal that you expect, if it is turn taking, if you expect, I mean, I'm not aware of any paper that suggests the appropriate metric or a set of appropriate metric, unfortunately. Your third point is the interpretation, how to interpret the metrics, right?
Yeah, more or less.
That is another open point, I think. Because what does it mean functionally that two brains share the same pattern? That can mean many, many different things. And yeah, we saw already that apart from studies that are, let's say, that have opposite results, okay? It might be that they have opposite result because they use different metrics, and also talking about synchrony is a problem because you don't know if you are comparing studies that actually did the same thing. So really, I think it's not only a terminological problem, it's also, I would say something that we should ourselves try to coordinate ourselves as a research community, try to coordinate better and do this effort, not to say, I do this, you do that, but try to, share the efforts and maybe explore different aspects, but try to avoid confusion. And also, because this confusion also affects the interpretation. Now you don't know if strangers synchronize more or less, because you don't know what is synchrony, so what does it mean? Fishes synchronize very well, but what does it mean in terms of physiological synchrony, of a neural response? Still, we don't know. So I don't have an answer, sorry. So for people that maybe this afternoon will join, we can talk about this more. So maybe.
Yeah, so thank you very much. So this is the end of this workshop, of the lecture part, but we'll still meet with the part of the team during the workshop. So I would like to thank you all for your participation, active attention, and first of all, of course, thanks go to European Union to make this event and grant possible. But personally, I would like to thank NIREX team to give us excellent lectures and workshops, and all the lecturers from our partnering universities, University of Trento, University of Heidelberg, and University of Warsaw. Thank you, our wonderful students. You were really great, asking really good questions. So I am really proud of you, because it means that, of course, you are great, but maybe there's somehow our involvement also to make you at least interested in science. And also big thank goes to Warsaw SYNCC-IN team. So thank you very much, Eva, Asia, Jarek, Maciek, Julia, and other people, Maciek also, for make this event possible. Personally, I am really, as an organizer, also happy from tiny things. Nothing exploded. So yeah, and I think it was really interesting. So let's celebrate. Thank you very much. And one more final word. I wanted to bring up our web page, but I was not successful at it, because I would like to tell that tomorrow University of Trento team is organizing very interesting online event. You can learn more at our web page. It will be dedicated to open science and all these great practices we would like to follow. So you are all, I guess, welcome to check and maybe sign in. It's online event, and it will be repeated. Yeah, it will be the first tomorrow, but it will be repeated. All the information might be found on SYNCC-IN web page under events block. So thank you very much again. And yeah, enjoy your 1 hour break. We all deserve, even we are a little bit behind the schedule, but we deserve 1 hour break. So 55. Now she's a bad policeman. Thank you very much.